an eminent taxonomist. So we welcome Dr. Prashant to this meeting. We also have other eminent entomologists who are also our directors, Dr. Varghese, Dr. Ballal, Dr. N.K. Krishnakumar, then our speakers, Dr. Dirk Ahrens and Dr. Peter Spick, because we always put Peter P-E-T-E-R, but his name is Peter Spick. Welcome you, sir, for this meeting. And also I understand so many people have registered. We have almost like 126 participants. It is going now to 127, including some of the deans, director assessors, associate directors, heads of divisions of various agriculture universities, ICR institutions. A big welcome to all of you, sir. I think we will have a wonderful discussion on this, and we will have come up with good recommendations at the end of this meeting. Welcome you all, sir. Thank you very much. Over to Sridevi.
Thank you, sir. Thank you so much for your brief welcome. And without much delay, we'll now get into the next speaker, expert remarks by Dr. Prashant Mohanraj, sir. And Dr. Prashant Mohanraj has worked in Andamans for 14 years, and he has opted to go there and worked on insect biodiversity and also trichogramma taxonomy. Then he has been transferred to NBIR, and he has headed the division of germplasm collection and characterization. And for a brief period, he was in charge director too. And he has several credits to his repute. But one significant achievement is coming out with the book in Hindi on butterflies of Andaman and Nicobar Islands. So that marks his credentials. And then now I request Dr. Prashant Mohanraj to give his expert remarks in this webinar.
Unmute.
Okay, good afternoon, everybody. Good afternoon once more. I will go straight into what I want to say. As we enter the Anthropocene epoch, and we notice the precipitous decline of insects across the world, this topic has been very pertinent to be discussed, the topic before us today. And I've been told that it's a mixed crowd that is present over here. So I will start with some very elementary things about insects and then move on to some other suggestions about the kind of systematic work that could be undertaken in the future. So basically, I'll be dealing with very briefly with the excitement and danger of collecting insects, the grueling work that goes into taxonomic research on establishing the identity of species, then the value of insects in museum collections, the changing functions of insect museums, the requirements for insect taxonomic studies, and finally, I will deal with a project that was in operation under the ICAR called the Network Project on Insect Biosystematics, which sadly came to a very untimely end. Now, the best thing to start with, I thought, would be with a master himself. So we start with Linnaeus, and the picture that you see here of Linnaeus is not the one that you usually see. This is the one that was painted of him after he returned from his first collection trip to Lapland. And then he wrote a book, a flora of Lapland, which was the first flora of a region from anywhere in the world. Faunas were to follow after that too. So he went to Lapland in 1732, and he collected mainly plants, birds, minerals, and also many things that were related to cultural aspects of the Sami people of Lapland. And he described about 100 new species of plants in that work. It is interesting to see that Linnaeus was held in very high esteem, and he figures on the 100 krona note of Sweden, as you can see, and the reverse of it has the Swedish king on it, or it had the Swedish king on it. And another little known aspect about Linnaeus is that his works were proscribed by the church at one point in time. In 1758, his works figured in the Index Librorum Prohibitorum, the prohibited works that were put forth by the church. So his works were banned for a while. And then these are the routes that you see of various students of Linnaeus who went to various parts of the world to collect insects. And the arrows show you two of his students whom he called apostles who came to India. One was Olaf Torren and the other was Karl Thunberg. Karl Thunberg is considered to be his best student out of the 17 apostles that he had. And so you can see that right during Linnaeus's time, India has had a head start in the description of species, and Linnaeus did describe about a dozen species from India based on the collections of his students. And finally, the last student of his, who is J.G. Koenig, also came to India. He stayed in India, he collected insects, and in fact, he wrote the first scientific entomological paper on an Indian insect on the termites of Tanjavur. So he collected insects too, and they were sent over to Linnaeus. So the point I want to make here is that in spite of our having such a head start over many other countries in documenting the insect fauna of our country, we still have a lot to do as compared to many other countries. And here you see that generally in India, we have neglected studies, traditionally, I mean, of nature, of plants, animals, which were not of use to us, as you can see in the first quote, where he says that the Lepchas are better than any other Indian who knows and cares for nothing about flowers or animals except those that he eats or that eat him. But this is not really true, because much before this, two people, Garcia de Ota, a Portuguese, and Henrik van Reed, another Portuguese person who were or a Dane actually, who were in India. They used the assistance of Indians who were familiar with plants, but these were only medicinal plants and nothing else. So it's not quite true that it's only those organisms that were eaten by us or that we eat that were of interest to us. There were medicinal plants too that were very well known. And in fact, Linnaeus used the descriptions and the diagrams from Van Reed's book and described them as species. And it is interesting that the Lepchas from Sikkim are very knowledgeable about their environment, and they had different names for different species of butterflies as also of birds. The bird identities have been documented. Unfortunately, the butterflies, nobody has done a correlative study between modern classifications and those of the Lepchas themselves. But studies on the birds have indicated that they knew their classification and ours match pretty well. So they had a very good knowledge of it. And these are birdwing butterflies. And this particular species was first seen by Alfred Russel Wallace, the famous biologist who, along with Darwin, propounded the theory of natural selection as a mechanism of evolution. So when he surveyed or studied the fauna and flora of Indonesia, on the island of Bacian, the first day when he landed, he disembarked over there, and then he saw a female of this butterfly, which is on your left. But it was flying very high and he couldn't catch it. He knew it was a new species. So every day after that, for the next 2 months, he went to the same spot and also around that area, trying to collect these butterflies. But he couldn't do it. But then after 2 months, he caught his first specimen of this particular butterfly. And this is what he had to say when he caught it. I quote it, I'm not quoting it in full. He has a much longer description of the experience that he had. What he says is that the beauty and brilliancy of the new birdwing butterflies that he saw are indescribable, and none but a naturalist can understand the intense excitement I experienced when I at length captured it. On taking it out of my net and opening the glorious wings, my heart began to beat violently. The blood rushed to my head, and I felt much more like fainting than I have done when in apprehension of immediate death. I had a headache the rest of the day. So great was the excitement produced by what will appear to most people to be a very inadequate cause. So that's precisely how I think entomologists feel when they collect species. Time, huh? Okay. Then when the Linnaean students went abroad to collect specimens, he used to write to them saying not to be afraid of exposing yourself to a certain degree of danger, because he hoped to reach a glorious, if anybody wanted to reach a glorious goal, they must take many risks. But this Linnaeus was saying when he was sitting in his home in comfort and in safety. And 5 of his 17 apostles died never to return to Sweden. One of their wives was so angry that she chastised Linnaeus for making her a widow and her children orphans. After which, Linnaeus never sent married people abroad. And these are some of the people who collected in India who died in various places. You know? And this is a slide that I thought I'd just show you to show how difficult it is to describe a species sometimes. This work was done by Dr. Pratapan, who was also a part of the network project on insect biosystematics for some time. He collected these beetles from across the country. He had to get specimens from so many museums from across the world and study the types and other specimens. And then after 10 years of work, he found that the beetle that was called the Polu beetle actually belonged to a completely different genus. He had to erect a new genus and describe a new species also. And this is a very large bee to show you the importance of museums in insect collections, which was collected by Wallace, and then in 1859 until 1981, no one saw a specimen till a single specimen was seen in 1981, which couldn't be collected. In 2017, it was thought to be extinct, but then it was rediscovered after an anonymous dealer was found to have sold it for over $9,000 US. Then the conventional view of museums is that they are very dusty, musty storehouses, and the job of curators is just to merely amass collections. But the modern view is that they are also scientists who have to produce scientific knowledge, and that has to be disseminated to the rest of the world. And they have to do original field work and research. And exhibitions are a part of the job of other people. Then going quickly through the requirements for studies, of course, we all know that we need specialized equipments like microscopes, including high-end photographic equipment, electron microscopes, then molecular techniques, of course, and for which we need expensive cryogenic facilities to store insects, insect parts, and DNA of insects. Then, as far as the future is concerned, it's already coming in. These are nascent technologies where magnetic resonance imaging and computer tomography can be used to study, especially insects that live within other organisms, like leaf miners or borers or parasitoids like ichneumonids and braconids. Without destroying the specimen, you can study the organisms within them. Similarly, artificial intelligence can be used, especially in the case of for identifying insects. And this is already being used in the case of birds, where the knowledge of birds is very well known across the world. Then digitization of specimens is, of course, very important. And what I'd like to suggest here is that there should be an integrated centralized national database of digitized images and their associated data, which should be accessible to all, even internationally. And we should rope in everybody, including the ZSI and other universities, which even house very small insect collections. And there should be funds provided to ensure that these collections are kept. Then separate funds should also be allocated for literature to be digitized, especially very old literature. And of course, now even the newer literatures are very difficult to access because they are all blocked with paywalls. Then the ICR should also make it mandatory, because we find that in many cases, when taxonomists retire from various institutions or when they pass away and there's no one else to take on that work, then those collections go to waste. So there must be some way in which these are either purchased or mandatorily transferred to some recognized repository of insects. And there should be a provision to purchase specimens from private collections too, and from institutions not coming under the ambit of the ICAR. It's interesting that the Danes, the French, and the British collected many insects from India, and they are maintaining them even to this day in very good condition. So it is important that we maintain our collections too. Linnaeus, why I want to talk about patronage is because patronage is extremely important, like funding. Funding comes from patronage. Linnaeus was patronized by the King of Sweden, and when he died, his specimens and his library went to his son. When his son died, it went to his wife and his daughters, and they decided to sell his specimens and the library. And an Englishman, a naturalist called James Edward Smith, purchased it for a mere £1,000, which is supposed to be a very cheap, a very low amount in those days even. And it is said that King Gustav III, if he had been in Sweden at that time, he was away in Italy and France on a tour, if he had been there, he would have seen that these specimens never left the shores of Sweden. And there is an apocryphal story too to that. Yeah, on his return, he dispatched, okay, I'm sorry, I'll move on. Finally, this is the network project on insect biosystematics, which ran for about 12 years. And it was at Delhi and at NBAR. It had a very long gestation period, but unfortunately a very short lifespan. And these are some of the selected achievements, like databases were there, which were very commonly used and very extensively used by students and research workers from universities and other places. Similarly, these are some other things. And I thought we'd just do a thought experiment. I'll end with this, that what would happen to a world without Linnaean taxonomy and taxonomists? As you can see here, there would be no organizing schema for the biotic world. And without taxonomists to unravel the building blocks of nature, many disciplines within the biological sciences, like biodiversity studies, conservation biology, and molecular systematics, would descend into chaos. Thank you.
Come out of the screen now. Thank you, sir. Thank you for that enlightening talk about different phases of the research taxonomic status and also the research that has been carried out earlier. I now request ADG sir, Dr. YP Singh, to say a few words.
Thank you, madam. Good afternoon to all. First of all, I congratulate. Am I audible?
Yes, sir.
Am I audible?
Yes, sir. Yes, sir.
Yes, sir. Very well, very well.
Okay then. So first of all, I congratulate Dr. Prashant Mohanraj for his excellent and I think the fascinating presentation, and he has given the overview of this development of systematics of insects. So now we start the Honorable DDGCS IACR sir, Dr. T.R. Sarma sir, and today's chief guest of this webinar. respected Dr. Bakbasalam, Director NBAIR, and the panelist, respected Dr. Ramamurthy sir from IARI New Delhi, panelist Dirk Erham from Germany, Dr. Peter Sipek from Czechoslovakia, and Dr. Swaminathan from MPAUT. As you know, that the taxonomy and the systematics are very interesting and fascinating field to explore the diversity of the organisms, as insects are the largest group, single group in the animals, and insect taxonomy has the wider scope of the application. Systematic documentation, description, cataloging of species help in the identification of this geographical distribution, which has the immense value in the agriculture also. So, as you know, that insect is the biggest group. Insects makes about 58% of the known global biodiversity and requires a large number of the taxonomists for its documentation. As Dr. Bhaktavasalam said in remarks that only 20% of insect biodiversity has been identified, and 80% is still unexplored. So, out of the 29 orders of insects, about 1 million species has been described, and 4 orders, that Coleoptera, Diptera, Hymenoptera, and Lepidoptera, that accounts about the 81% of the described species. So this insect taxonomy is the basic science for is very crucial and linked to the other disciplines also of entomology, like the pest management. Either it may be the biological control, ecological, physiological, and many more comes under this also. So without the proper identification, it is very difficult. I think the most difficult to manage the pest. As you know, this papaya mealybug in India that makes the havoc, and after the proper identification, this management could be done. Taxonomics collection provides the permanent record of this historical distribution of the species, prediction of extension of these species, pest and disease prevalence, and the insect importance as the new species invasion also. So the species taxonomy will research as a advanced scientific this discipline, as I think so, with advanced technologies like the TAM and SAM that is coming forwards. So fortunately, present day taxonomists are equipped with the molecular tools to make the identification very quick and authentic. Earlier, we have to take the identification with the, I think, much time we are taking. Now it is very quick and authentic also. The young and building entomologist, I think, should take the taxonomy as the career and describe the untouched group of the for the benefit of this science of this entomology. With these some words, I congratulate NBIAR for taking initiatives to organize an international webinar and insect systematics, and hope that deliberations will be highly fruitful and will come out with solid recommendations which will serve as the roadmap for insect taxonomy. I think I thank the organizers for inviting me in this webinar, very important webinar. And here I stop and thank you the organizers and as well as all the participants for this webinar. Thank you very much.
Thank you, sir. Thank you so
Dr.
much for
Sridevi, before we proceed, I want to welcome Dr. Manjunath, one of the senior most entomologists and biocontrol expert. Sir, welcome, sir. That time I didn't see you. Welcome, sir.
Welcome, sir. Good afternoon, Manjunath, sir. Sir, thank you for your kind remarks, Singh, sir. Now I request our chief guest, Honorable DDG Crop Science, Dr. Sharma, sir, to give his remarks and opinion, sir.
Hello, good afternoon, good morning, and good evening to all, because you are sitting in different corners of the country. Honorable former DDG Horticulture, Dr. Krishnan Kumar ji, my former colleagues, Dr. Chandis Balal, former Director NBAIR, and former Director Dr. Varghese, and others, my present colleague, Director Dr. Waqt Vatsal, Dr. Baipi Singh, ADG FFC. I could see Dr. Manjunath here and Dr. Prashant Mohan Raz, different distinguished speakers, ladies and gentlemen, participants. First of all, I would like to thank you all, particularly the organizer, for inviting me here for this particular webinar on insect systematics, its importance, challenges, and way forward. Let me first tell you that taxonomy is my one of the favorite subject, but if we see in present context, all taxonomists are being considered as an endangered species. No one would like to take this as a subject. That's very unfortunate part. Even at country level or national level, we always think in different meetings and at different platform that how to revive this subject, how to sustain this subject, because we know that without knowing the identity of an organism, whether it is insect, microbe, fungi, plants or animals, we cannot do systematic studies. So that's how the significance of this particular webinar becomes more important. And I was looking at the profile of speakers, and I'm really very happy to see that world authorities on taxonomy have been invited for this particular webinar by the organizers. And I'm sure that their lectures will inspire the new generation of scientists to take this as one of the important subjects in biology. Our institute, NBAIR, is one of the very important institute of ICAR, and I think this is the fourth or fifth time I'm interacting with the scientists of this particular institute through various meetings, although I did not get a chance to visit the institute as yet because of COVID-19 situation. But definitely whenever I will visit Bangalore, I would like to see this one of the most vibrant and important institute of ICAR. For today's webinar on insect taxonomy, we would also like to see that this is a subject which is evolving very fast. Not only morphological taxonomy is important or traditional taxonomy is important, but molecular taxonomy has to be integrated with the ongoing programs. And I am happy to note that our institute, NBAIR, is actively working on molecular taxonomy. Looking at the literature, when you see not only now the traditional DNA markers which we use for the identification of insect like the cytochrome oxidase 1, cytochrome B, 16S, 18S, and elongation factor 1 alpha genes. But also, one can use now whole genome sequence information for some of the important species. I'm sure that you might be aware that any organism which was sequenced fully or whose genome was fully decoded was Drosophila melanogaster. The genome was published on March 24, 2000 in a very prestigious journal Science. It's a small genome. And you know that Drosophila is used as a model species for studying various human disorders. And if you read that paper, its sequence size is around 120 million base pairs, which is, I would say, very small among eukaryotics or eukaryotes, except fungi, where average genome size is around 36 to 40 million base pairs. But being a small genome size of Drosophila melanogaster, this was the first organism used for decoding of full genome of this particular species by the scientist in an international consortium. And in this particular genome, more than 30,600 genes were predicted in the genome sequence of this Drosophila. Many of these genes are present in gene families with different copies and copy numbers. So genome sequence provides an important information to all of us for understanding the molecular biology of an organism, particularly insect. Just now I was looking at the NCVI database. Those who are biologists, they are familiar with this NCVI, National Center for Biotechnological Information, situated in US. More than 1,219 insect genomes have already been sequenced. But the question comes now, how many sequences we contributed from Indian side? I'm not sure we have done any complete genome sequence, or I am not aware if that is done from India. So being a premier institute of Indian Council of Agricultural Research, where we have many taxonomists and molecular biologists at Bangalore, we should think of sequencing at least some of the important genomes of some of the important insect species which are relevant to us in India. And there are very important insect genome databases wherein one can do analysis of the sequence for understanding their phylogenetic relationship and also evolutionary biology of insect species. In spite of the fact that we have all molecular techniques available with us nowadays, the role of a typical taxonomist cannot be ignored. As Dr. Prashant told, we should try to retain our taxonomists in one or other form in the system, or they should see that a new batch of scientists should be trained in this very important field. If we think of counting taxonomists not only in entomology, even in plant species, we can count on our fingertips. It means they are very limited in numbers. And whenever such proposal comes for funding in Department of Science and Technology, where I chair a task force on funding, we always give preference to the taxonomist. So one should not say that we are not getting funds to do taxonomy. Everyone realizes the significance of this very important field, because as I told in the very beginning, without identification of organism, we can't do anything. We cannot devise any management strategy. We cannot understand its biology. We cannot understand the biodiversity. Therefore, identification is the key step for the analysis of an organism, for which now barcoding is being emphasized for many of the organisms and by using all types of DNA markers. And for gene expression analysis, transcriptomics is being used. As told by Dr. Vatsal, I started my career at IRI from sequencing of plant genomes. And Dr. Krishnan Kumar knows very well that during his tenures, we were sequencing mango genome. And I can't forget that particular meeting in which within 1 hour, he sanctioned 2 crore rupees to add to the existing projects. That type of decision making and interest in science is very, very important. I was hardly principal scientist at that time, working with a very vibrant group of genome sequencing in rice, pigeon pea, tomato, and that was mango was our other target, which comes under the domain of horticulture. So in a single meeting, 2 crore rupees, can you imagine? That's to an ICR system. It needs courage. So not only we have limited number of scientists now, those who are contributing to the latest advancement in science, but we have very few administrators, science administrators, who takes quick decisions to understand the significance of such approaches and techniques. So until unless we train next generation of scientists in these techniques, we cannot move ahead. In that context, organization of such webinars, and now it has become more easy because of this electronic format, we can spread our message to the large number of people in the shortest possible time. And I can see 173 people, those who have joined today in this webinar. Had we thought of doing this particular seminar physically, it would have cost us lakhs of rupees. And probably we would not have the privilege of listening to our scientist colleagues from abroad, particularly Dr. Dark and Dr. Peter, and other colleagues, those who are speaker in this particular webinar. So COVID-19 situation has not only created certain barriers, but has also given us a lot of new innovative approaches to interact. And this is one of the very important symposium or webinar series, which I would say one must attend. And I am not aware what Dr. Prashant told, that there was a project on insect taxonomy and which was closed down by ICAR. I would like to request Dr. Vatsal, why don't you come up with your proposal and file? Where is that? We should dig it out and see that suitable resources can be allocated for this particular field of science.
Yes.
So my presence here is not only to speak what I wish to say, particularly on molecular taxonomy, because molecular taxonomy was started somewhere in 1994-95 as a funding source by the Department of Biotechnology, Government of India. And I got funding under molecular taxonomy project as an assistant professor. So it's an old field, I think more than 24 years old. Funding has already exhausted. And everyone realizes the fact that we need to support this particular branch of science. But still we see that people don't have interest, which we are unable to create among the new generation. But it's very interesting field. If you study by using latest approaches, by using scanning electron microscope, I told Dr. Vatsal that it should be there at your institute. If not, then why don't you put it in your EFC? Now we have tabletop 10. Why don't you purchase that? So if you use those techniques, those latest equipments, you can make this particular field very interesting. So it's a very, very interesting field because I personally like this particular taxonomic work, whether it is plants, fungi, animal or bacteria. And I'm sure that today's webinar will definitely throw some important lights on this subject and shows new ways of doing taxonomic work in case of insects. So my best wishes to all of you. We are already 2 minutes late, so I don't want to take long, and I wish that this particular webinar should be one of the most successful webinars, and you should bring out very clear and crisp proceedings, not more than 1 page, okay, and recommendations which we can think of including in our ongoing programs. And luckily we are preparing the EFC of institute and also of the council, so that will give us an opportunity to include some of the important areas and allocate resources according to its significance. So again, best wishes to all, and congratulations to the organizers. Thank you very much for inviting me here. Thanks a lot.
Thank you, sir. Thanks a lot for your kind remarks and also wisdom, expert opinion, sir. We'll be certainly bringing out the proceedings, sir, as you have suggested. And then
Dr. Sridevi, before we go for vote of thanks, I want to just tell the views of, the achievements which we have done from NBAR. Sir, already we have done whole genome for one insect, sir, that is the Lucinodes arbonalis, that already we have submitted for publication. One more insect also, it's almost to be completed, sir. One leafhopper, Amraska.
Are you there in Earth Biogenome project of?
Yes, sir. Yes, sir.
Okay,
yes, sir.
Very good. Very good. Very good.
So this biosystematics project.
Not 1 or 2, now hundreds, thousands.
20 we have proposed, sir.
You should have proposed 100.
Yes,
Now the technology is very cheap. So,
okay, sir.
anyway,
that biosystematics proposal I will again discuss with you, sir. We will send.
Yeah, we have to revive that proposal.
Yes, sir. Yes. Thank you. Thank you, Rupert. Thank you.
Thank you, DDG Crop Science, sir, and thanks to our director, sir. And now I request our head division of germplasm collection characterization, Dr. Sunil Joshi, sir, to present the formal vote of thanks. Sir, you are not audible, sir. Sunil Joshi, sir, you are not audible. No,
Joshi,
sir.
you are not audible. You are not audible.
We are not able to hear you, sir. Joshi sir, you just see, check what is happening. Ah, he went. Sunil Joshi sir. Meanwhile, sir, to DDG Crop Science, sir, I would like to put forth that we are having collaboration with Dr. Dirk Ahrens, sir, from Germany. And we under the NBA agreement, we have taken Form B, which has been facilitated by our directors from NBIR, and we are able to carry out the collaborative work with Dr. Dirk Ahrens. And he is all-round renowned taxonomist working on sericini and tribe sericini. We are doing it. And same with Dr. Peter Sipak, we are trying to establish the collaboration in Prague, who is in Prague, on the immature taxonomy, sir. So this good. I would like to put forth before you, sir. That we are in collaboration, and Dr. Dirk Ahrens is identifying many specimens on Indian sericini. I just come out with the monograph of Indian sericini in 2016. And he is helping us in identification and also building up of our referral collection, sir, over here.
Okay, very good. So if Dr. Sunil Joshi is unable to connect, Dr. Sridevi, you can propose vote of thanks. So it's only a formality, doesn't matter.
Yes, Dr. Sridevi, I think you go ahead.
Because he is unable to connect.
He is unable to connect.
No, we are trying, sir.
Okay. Now we can hear.
Yes.
Dr. Joshi.
Yeah. Thank you.
Yeah.
Am I audible now?
Yes,
sir. Yes, sir. Please.
Sorry for that. I will start my vote of thanks now. Our most valued invited guest from ICR, Honorable DG, DDG, ADG, Dr. Dirk, Dr. Peter, our former and present directors, Dr. Manjunath, ladies and gentlemen. It is my privilege to have been asked to propose a vote of thanks on this occasion. On behalf of our whole institute, I extend a very hearty thanks to our Director General for giving us the opportunity to hold this international webinar. We are very much thankful to today's chief guest, Dr. T.R. Sharma, Deputy Director General Crop Science, ICR, for continuous support and advice which have greatly helped toward the successful organization of this event, and for kindly accepting our invitation and gracing the inaugural function. Thank you very much, sir, for your suggestions for reviving the NPP project, sir. Our thanks go to today's guest of honor and a person who is very close to us, Dr. Y.P. Singh, Assistant Director General FCC, ICR, for his valuable suggestions, guidance, and making himself available for this program and for his remarks. We are grateful to our director, Dr. N. Bhaktavasalam, for constant encouragement, moral support, and his inspiring mentorship. We express our great sense of gratitude to our former head of division, Dr. Prashant Mohanraj, for giving his time and for agreeing to join us for this meeting and for delivering such a wonderful expert remarks. We are highly delighted to thank Dr. Dirk Ahrens, ZFMK Bonn, Germany, who is a renowned taxonomist in Scarabody with special reference to sericini, and to Dr. Peter Sipak, Praha, Czech Republic, who is a taxonomic expert of Citonini, for giving their consensus to be speaker on very important and highly useful topics. We sincerely thank our close associate, Dr. R. Swaminathan, professor from MPUIT Udaipur, for readily approving to be part of this webinar in spite of his very busy schedule. We are extremely thankful to our guide, Dr. V.V. Ramamurthy, Division of Entomology, IRI. Unfortunately, he was not able to make it today, but who was instrumental in building this webinar, his constant support, guidance, and encouragement. Our special thanks are to Dr. T.M. Manjunath, who is our constant support and who immediately agreed to be part of this webinar. We are thankful to you, sir. And now our ex-directors, Dr. N.K. Krishnan Kumar, because of whom many young potential taxonomists were brought to this institute. Dr. Abraham Varghese, who almost forced many of us to work on taxonomy, and some of us have turned into very good taxonomists today. Dr. Chandishar Ballal, whose constant determined efforts helped us to cross the barrier involved in international exchange of specimens, and because of which many taxonomists were benefited. We will always remain thankful to you, Dr. Krishna Kumar sir, Varghese sir, and Chandish ma'am. We are thankful to deans, associate deans, heads of divisions of different state agriculture university for giving their valuable time and for attending this webinar. We acknowledge the unwavering support received from the HODs, staff member, administration of our institute, and AKMU for logistics support. Without Dr. Pratipha and her staff, conducting such program wouldn't have been possible. We are thankful to young enthusiastic taxonomists from NBAIR, IARI, UAHS, ZSI, and Model College Pune for coming here, and I expect a very good input from them. It is our pleasure to host all the participants for this international webinar on insect systematics. We are thankful to all of them. Last but not the least, Dr. Sridevi, who skillfully managed last-minute invitations, 11th hour changes, every minute additions in registration, non-conforming guests without losing her cool. We all are thankful to her for taking all this responsibility and efforts put in for our division. Thank you, one and all.
Thank you, sir. I place on record the thanks to our head, sir, Dr. Shunil Joshi, sir, who is supportive and
Sir, I think NBR is having internet issue. In morning also we faced this issue in trial also, sir. So they have made me a host. I will try to rejoin everybody, sir. Please give a minute.
Available. डॉक्टर टी.आर. शर्मा जी नमस्कार।
नमस्कार, कैसे हैं आप?
बिल्कुल बढ़िया सर। Having a small meeting on phytoplasmas.
Oh, very good.
5th February, and it will be a one-day meeting just to take all experts with the 30 years.
Where you are organizing?
It will be virtual meeting.
Okay.
But it is in collaboration with the Wood Science from the Ministry of Forest.
Okay.
And Entomological Society of India.
I see.
Persons from USDA and others are also joining. Professor Viraktamat will be giving a talk on leafhopper, plant hopper vectors.
That's really good.
So
yeah, phytoplasma is also very less studied organism in India.
Very less. Nobody is literally.
Yeah.
That's one of the reasons why we took it, because we find that it is becoming more and more invasive, and throughout the world the vector status is not clear. So we thought we will. I will send the link, and in case on 5th February.
Yeah, sure. If I am free, then I will try to join. Is it in morning hours or evening?
It will be from 10 to 5 because about 10 speakers.
But morning we have SOC meeting, so probably I will be able to.
Yeah. Anytime. I'll just send the link if.
Yeah, sure. So I think we lost the link. So now I would like to take leave and thank you very much. Thank you all. Namaskar to all.
Thank you.
Before you take leave, I just want to, since you raised this issue, please, now that I am out of ICR, I can speak a little freely. The systematics network was going exceptionally well. But for some reason, Dr. Sandhu could not appreciate it in the way it should have been appreciated. As a result of which, it could not go forward. But the work that was carried out was absolutely outstanding. And we tried to defend it for a number of times, but it is difficult when at some point very powerful bottlenecks take place. So I think you should probably support this one network with adequate contributions coming from the molecular systematics side, because it should not be only one-way traffic. And together let them work on molecular and traditional systematics and take it forward.
I would like to see this whole document. Where is that document? And we will see. We will try to see isko funding mile.
Yeah, I think Bhaktavatsalam and others, Dr. UAS and NBIR were heading that particular group with NBIR as a lead. But outstanding work was done. Probably I can arrange, and when you come to Bangalore or something, you can have a look, and I'll also try to arrange it from the Bhaktavatsalam and also one from Dr. Viraktamat who was heading it.
Yeah, sure. We will do that.
Sir, I just thought I'll flag this issue, but for some reason it was bottleneck was there.
Okay, sir. Thank you, madam.
Thank you, sir.
Palal, thank you very much.
We appreciate your talk, and
Thank you, sir.
very beautiful words spoken with taking from traditional to how it should be from the other way side. The reasons we took Lucy nodes was we were permitted by ICR when I was a director to identify only 2 insects, not to go beyond. We thought that though, brinjal shoot and fruit borer was one of those insects which was defying all logic, both in terms of resistance as well as breeding for resistance. So, and being an Indian insect, we told them to sequence it to start with. But the times have changed. Now they should work, as you said, on hundreds of insects.
Yeah, it's very cheap and rapid technique now.
Yeah.
So one can add many number of species.
Yes, sir.
Okay, thank you, sir.
Thank you, sir. Thank you.
I'm
Thank you,
very
sir.
sorry, sir. In between, there was a trip in the internet connection at the institute. We could fortunately join back. Thank you, sir. Thanks for all your valuable. Sir, if DDG sir and all permits, we can go for the technical session.
Ma'am, just now DDG sir left, so we can proceed with the program, ma'am.
Yes. Okay.
Shashank, you should provide that entire background to DDG about that systematics. You please coordinate, since you are aware whatever that happened.
Okay, sir.
So talk to Dr. Ramamurthy and yourself and to Dr. Bhaktavatsalam and put it up to DDG.
Yeah.
Okay.
Sir,
Okay,
so
sir.
that
we start
we
our
should go forward and don't wait for things to beyond a point. You have to heat the iron when it is hot.
Yes,
start
sir.
the
Yes.
technical session, sir.
Okay, sir.
Okay, sir. Thanks to all of you. We'll just start our technical session. We have a very renowned speakers with us, and it is my privilege and delight to introduce the first speaker of the technical session, Dr. Dirk Ahrens from ZFMK, Bonn, Germany. Dr. Dirk Ahrens is heading the division of Arthropoda, looking after the Coleoptera section at ZFMK, which is popularly known as Alexander Koenig Museum in Bonn, Germany. His research interests are evolutionary biology and ecology of beetles, with particular interest in chaffers, that is Scarabadia group. Dirk has worked on dung rollers, then onto the tribe Sericini, and he's working on molecular and morphological phylogeny, morphometrics, evolutionary biology, comparative phylogenetic analysis, integrative and classical taxonomy. and DNA taxonomy, along with biogeography and taxonomy of sericini across the continents. His particular interest on South African continent has come up with a very good publications, and he has worked in several museums: Schenkenberg Zoological Museum, Dresden, Schenkenberg Entomological Institute at Berlin, and also he was a consultant for Helvetes, which is a Swiss development agency, and he has worked in the Zoologic Staatssammlung in Munich, and also Natural History Museum, London, and presently he is heading the division of Arthropoda at ZFMK Bonn. He has several publications, about 190 on scarab taxonomy and phylogenetics, as well as on morphometrics, comparative phylogenetic analysis, and DNA taxonomy in journals of international repute. To mark his contribution, there are several revisionary studies, especially on genus Tetraserica, and also his contribution on a monograph of the sericini of India that has come up along with his wife, Sylvia Fabrice, 2016, is a monumental work and also a foundation for the Indian sericini to take forward. And both Dr. Dirk and Dr. Sylvia has done a wonderful work on scarab taxonomy, and we will be delighted to hear your talk, Dr. Dirk, and you can take over and present your talk now.
Okay, thank you. Thanks a lot for the invitation to be able to speak here. I will try now to share my presentation.
Yes.
So, okay, here we go. Okay, so yeah, I was pleased to give a short more or less, an introduction on integrative taxonomy, and maybe help to provide an outlook for the future of taxonomy. All this kind of view comes up from my past 20 years of experience as taxonomist, trying to learn the new methods to implement it in my work. So here we go. As I don't know exactly what kind of audience all our participants are, I mean, I start more or less from the basics. So just saying quickly some corner points of what is dealing taxonomy with. So taxonomy is actually the science of naming, describing, and classifying organisms, including all the different kinds. And yeah, as already mentioned, the species name is kind of the most important information we have in all of our knowledge and databases, where the information is linked over for the different, huge different numbers of applications of taxonomic information in the science. So it goes from medicine to nature conservation, the public, farming, pest management, etc., etc. So the importance of taxonomy is enormous, and so is our work we are doing daily. So taxonomy, what it is actually, I mean, of course, more or less everybody is dealing with taxonomy, and but the precipitation is also to develop taxonomy as a science. We must be aware that taxonomy includes two separate processes of knowledge gain, and also where hypotheses are tested. The first thing is that taxonomic names are going to be set into an existing reference system of names, so by their IDs, according to the principle of priority and the Linnaean nomenclature. So we have the type specimens, which are the basic fundaments of our whole taxonomic efforts, and the nomenclature rules for zoology or botany. And these kinds are the guidelines for defining more or less some roads where we should move. But then as a second process, and this sometimes we forget about, or I mean, not really if we forget, because now there are a lot of more modern developments in this direction, is the delimitation of the species units itself. So the question is how we define one species and how we define the other species. So these are the big questions. And the development of taxonomy is more or less in this part, in order to modernize it, to make it faster, etc., etc. So this kind of delimitation of species units, how we define the species, which concepts we use, which methods and which kind of data are the crucial questions in taxonomy today in order to achieve our goals. So, yeah, in all of this, the species concepts have, of course, the very important role in the questions how we define species and how we recognize them. And I mean, current theory shows up there, of course, can be different species concepts, and you all are aware of that. Discussions about species concepts are going on for more than 100 of years, and and still people are arguing which would be the best concept, but actually in the way of speciation and different traits and character systems are like going into distinction between arising two species. And so not all the characters are contemporarily evident from two different pairs of species. So that's why there can be also discrepancy between the outcome from the comparison with different species concepts and also the outcome of the whole process of identification, or the number of species found can be different. So, but in all of this, we need to consider that the species concepts also in some way look a little bit on the way of, or the mode of speciation, rather than the definite outcome, so like evidence of speciation. And of course, the final outcome of any speciation is a genetic diversification, so that every species is genetically separate by another. Of course, in very young radiation and speciation processes, this is not always the case. So that is what makes taxonomy so difficult, and where we have to deal with. So actually the topic of my talk is what is integrative taxonomy. So integrative means they are coming different things together, and if we debate still on different concepts, but what people are agreeing is that there is a hierarchy of different types of character and criteria used for species delimitation. Evaluating different lines of evidence in a formalized framework is what would probably makes our species hypothesis more robust, and maybe also sometimes faster to achieve a result in species identification. So actually there are, in current literature, there are more or less two strongly diverging concepts of how integrative taxonomy is being used. So that, for example, we use different sources of data, we analyze them either in a separate way or in a more iterative, or in a single unique analysis. And then in these two different results, we get in concept one, we get for each of the data sets different results, but then we have to sort out these results by criteria of congruence, of cumulation, of consensus, or of candidate species approaches. Which of the results would now in the end apply to a decision to make if two species are distinct or not? In the second concept, what is more or less an iterative taxonomic way, because there's just a single analysis which takes into consideration different sources of data, where in the end we have just a single result. So this kind of concept would probably be more preferable, because in the end there's no need to argue further with subjective arguments to which result we come in the end. So in this kind of framework, also this famous taxonomic circle scheme was developed by Rob Desal and co-authors, where they have graphically shown how the different traits of characters would come together in order to make a decision whether two taxa would be separate or not. But however, for example, if we take the classical way where the taxonomic basis is based on a geographical pattern of two separately geographically occurring populations, according to their opinion, if then this difference of geography is supported by morphology, then we would come out with a species hypothesis. Or if we do have such a geographical distinction, no evidence in morphology, but in DNA, then we would have a species evidence. But in this way also populations would be characterized. So from the very beginning, these principles of integrative taxonomy always had also kind of problems in their assumptions that we need to be able to distinguish differentiation between populations and differentiation what we find between species. So even if we have multiple evidence, this kind of recognition of where is the species boundary is really one of the most important. But of course, I mean, as we're facing many requirements and tasks to taxonomy, we need always also to take reference to for what we trying to modernize taxonomy and what needs are there for taxonomy. Of course, for example, there's a pest management, there's a global diversity crisis that we know that many species going to be extinct by the destruction of habitats and so on. So there's a need of rather quickly characterizing the huge diversity we have on Earth. But at the other point, we have also the need to clarify between problematic species where different hypotheses are competing. Like, for example, in many large vertebrates, we're now coming up different hypotheses of that we have in front of us distinct species. So, and in these cases, we need proper methods and also reproducible methods by which we can say this is one species or these are more than one species. So we actually have to adapt our doing to what we need. And that also just having the kind of framework in head that we would like to characterize global biodiversity. Of course, we need to have in mind that we have then to have a complement and a complete reference set for all of the species for at least one trait that we are able to say, okay, this is a new species or this is not a new species. And currently we have an integrative taxonomy, the tendency that we add more and more character traits like 2D images, morphometric data, barcodes, genomic data, or in future we will have CT scans or 3D images, genomes. But even if the information content of these characters increase, the number of species for which we can or we have raised those data is rather limited. So this is the axis down here, the number of taxa we have kind of evaluated for them. So but what we have is kind of Linnaean style descriptions with morphology, with 2 lines of words. And what currently taxonomists are trying with by hand of their taxonomic revisions is to upgrade those old school descriptions by better descriptions with more characters in text, or by 2 million nowadays descriptions where we at least have at least 1 page of text and images, for example, insects, for example, images of genitalia or whatever other 2D photographs. So and this is kind of the one of the big tasks to expand this kind of morphological knowledge to all species. So that's one field of work. So, but at the other hand, we have other very valuable tools like DNA barcodes, what is also very available and very easily to extract. And so, I mean, now in the next slides, I will kind of trying to discuss also on which road of characters we should proceed. So, for example, why we would use morphology for the characterization of the organisms. Of course, the physiognomy and morphology of an organism is already saying quite a lot of the lifestyle, about the biology, and without morphology, we would not be able to implement the knowledge of the fossil record, which then helps in counterpart also to date molecular phylogenies. But morphology is also one of the most complexest character traits in organisms. And of course, morphology is much more easily accessible to our own kind of categorization and our understanding of categorization of things. Like, for example, if we have here a barcode of a certain species or the morphological image of this image of a morphology, of course, we would all agree that it is more easy for us to recognize the species by the image of morphology rather than the barcode, which is not comprehensible for our understanding. For a computer, it would be probably, both the ways possible. But morphology has in this way also for the communication an important role. And it's of course also accessible by distance, like for example in larger mammals, birds, we can collect data by just visual observations, and we can use them also then for computer-based analysis. Of course, we know all the data elaboration and morphology like it was in the first place, like by text, by words, by dichotomic keys, which was then later on substituted and extended by dichotomic keys and images, or even then interactive keys on computer illustrated also with very good images and so on and so on. So this was one of the first steps how to expand those morphological data sets to all species, so that our possibilities to improve the success of identification of animals. But of course, morphology has also problems. So sometimes the recognition of two states of characters is difficult, especially when those two characters overlap in a certain degree. And there comes in the problems of subjectivity in terms of recognizing species. And so researchers in the past have tried to objectivize this and to create and generate ways of analyzing data and refining those character states by hand of quantitative methods like distance measurements, landmarks, or shape outlines, which then were computer-based analyzed. But problems with these morphologically and even quantified characters is always that there can kind of overlap, and the outcome of such analysis depends often on the way how we initially define our species assignment. So the statistics behind finding significance between distinguishing two forms depends often by our initial specimen assignment to certain taxa. And this becomes even more difficult and evident if we try to, for example, using morphometric data in order to use kind of model-based clustering in order to infer the number of species we find in a data set, which maybe we can then verify also with molecular data. So that makes things more difficult and complex. And as you can see from those curves from model-based clustering, that there is not such a clear hypothesis visible that there's the kind of that we have one or clearly two species or four, but the difference is because of the amount of overlapping characters, even in morphometric data, is quite enormous. And this can lead also if in more advanced linear discriminant analysis, where we try to reassign specimens to certain samples and species, can be then prone of errors if the characters are not clear enough, distinct. So, and this can go up to that point that even between well-recognized species, we can have a kind of large amount of overlap between different hypothesized species. But if we choose, instead of hypothesized species, we choose kind of mitochondrial defined lineages in terms of larger populations, we can also see that morphometric characters in the same way correlate and are distinctive between different populations. This is what I already mentioned in the beginning, that in the end, the difficulty is not to find differences, but to give these differences a meaning that they are being assigned with a species boundary. So, and in this all comes then this other character trait, which, as we are speaking about integrative taxonomy, you of course want to see if we use other character traits, if this problem can be overcome. So why would we use DNA for morphological classification? Of course, DNA is a universal character system. We can identify it for all developmental stage, and so we can use it also to identify food or host plants, prey, and we can kind of assign different life stages to the same kind of species, what by hand of morphology would be kind of difficult. And it is kind of easy to analyze, and you must not be a specialist of a certain group. So DNA has its values in species taxonomy. And nowadays we have kind of very good databases like the BOLD data system for barcoding, where you can sequence a barcode of a species and then search in a query the reference database in order to identify your samples. This kind of DNA allows quite easily to make species assignments based on no prior knowledge of the species identity. So you can have tree-based methods in order to assign your species samples to a species, which there are different methods, which I will not treat here in detail. Or you can use distance-based methods where you assume a kind of barcoding gap, and probably lots of people have heard already about this. But in fact, the last 15 years of barcoding empirical data have shown that such a assumed barcode gap does not really exist for the most of the species, especially if you then analyze the whole geographic range of a species and of different species. So the more deeper we going to sample our specimens, the more problems we're having to use this kind of distance-based approaches. So that's why also other methods have been developed, which are more or less similar to morphology, are based more on the fact of revalidation using fixation index or the multi-species coalescent, in which you're having a certain species tree, and then the different nodes are investigated, whether according to the multiple species coalescent by the gene tree, the separate gene tree phylogenies, have then an agreement and posterior probabilities for certain nodes, which are then kind of deciding whether this is a good species or not. So for example, in this case, the red ones would be no splits between specimens, and the green ones would be different splits between species. But of course, things even with molecular identification methods are not that easy, because the speciation models and the models of species inference need additional priors to know about the speciation itself, which is the population, effective population size, but also the phylogenetic depth of the characters. And these variables, normally we don't know for each species. So we assume them to be similar between the different species, but in fact they are not. So there are coming problems, which I later on will be summarized in a separate way. So, but when using DNA, as already mentioned, there are a lot of more pitfalls and problems. For example, if we use a different sampling of specimens, there can be different results of species inference. For example, this study from 2016 have analyzed different clades of chavers in order to infer species, and we did at one hand the whole lot of species, and then we analyzed separate clades like those which are marked here with letters separately. And as you see from the outcome by these columns here in the species delimitation, there is no congruence. So that means if you're using a certain sampling, the result can be one. If you use another one, the result can be different. And in these blue bars on the side, we have just mapped the effective population size, which we have calculated based on the barcode data. So there are strong differences, and so that means these kind of strong differences between the species and the estimation process of species boundaries can cause serious problems and inaccuracy. And of course, also text bias dispersal can influence the result of species delimitation, and as well also the choice of the genetic markers. For example, if we have for these Pachypus species, where the females don't fly, they don't have at all wings and elytra. When we use CO1 and 16S, we have kind of huge numbers of species like 60 or even more. If we use nuclear genes, the number of species we obtain are much less. And also, according to different methods, what we use when we cluster morphology or using GMIC, so tree-based methods or coalescent-based methods, we're getting different results in species delimitation. So all of this kind of problems, especially also when we're using just CO1, so mitochondrial DNA data, we must be aware we get problems because of spatial paraphernalia. So it was estimated that up to 23% of all species are not monophyletic, and also Wolbachia infections. So these are microorganisms and unicellular organisms which affect the cells and having a kind of impact on the heredity of the mitochondrial genome, and they lead to bias in the diversity of haplotypes, and bias those results of barcoding. Of course, then also mitochondria have a low recombination rate, so the mitochondrial data can show past geographical patterns which current, like Pleistocene refugia, which nowadays are not present anymore, but we can see among the data kind of divergence. And mitochondrial DNA obtains or reaches 4 times faster coalescence nuclear DNA. That means that when we use mitochondrial DNA for species delimitation, we of course see much more clear clusters than with nuclear DNA. And also, and this was from the beginning not so clear, that in many cases, especially in insects where we can verify morphospecies also with the shape and morphology of genitalia, CO1 data lacking resolutions. So, and of course, there are already, as mentioned, other problems when we use DNA-based methods, like the variation of effective population size, which is one of the priors in all tree-based and coalescent-based species delimitation methods. As already mentioned, dispersal activity plays a crucial role. So we have between different organism groups, different amounts of a barcode gap, and also between intraspecific variation. Then also the depth of the tree, or as mentioned as taxon sampling, plays a role, and the structure of sampling itself, like if it is balanced between the species or unbalanced, the number of singletons, this plays an enormous role. And as well, the idiosyncratic gene evolution. So that means every gene kind of not always is being under selection, and those they develop neutrally. And so every gene tree can be different from the species tree, and that's why we should analyze as much as genes as possible in order to infer properly the gene tree. Yeah, and with our sampling choices and choices of pairs like theta, we have also in molecular analysis a quite huge amount of subjectivity. So this is what always was thought it is not there, and which was one of the major arguments against traditionally taxonomic made taxonomy, because they said it was all subjective decisions made in everybody's taxonomist head. And yeah, and with the most kind of fashionate method, the multi-species coalescent, which is the most representing the highest state of the art using multiple genes, even that method has currently problems as it divides structure and not species. As I already mentioned, the difficulty between this distinct between populations and species. So, but last but not least, there are also other, of course, other ways of evidence to infer between different species. This can be ethology, this can be even ecology, and recently also niche modeling is used oftenly in integrative taxonomy. For example, deriving from the distribution data of different species a niche model, and in this niche model, if, for example, in these two cases of different gecko species. They indicate different potential occurrence areas, and then they are used as a supportive argument for species inference. But this kind of data from distributional on the distributional ranges are of course inferred by prior species IDs with other evidence, and so this kind of inference using niche models for species distinction is kind of circular, and we can really use it as a own standing proper argument for distinction of species. So there are a lot of problems with all the kinds of different character and traits, and that's why it makes probably more intelligent to use an integrative approaches in order to infer information from other data sources in order to get to the truth. And of course, as a curator, and also about as also many of you are dealing with entomological collections, the role of museums in taxonomy and also for integrative taxonomy is quite important, because museums are treasures of biodiversity in time and space, and many museums worldwide contain unidentified specimens which can still be used to access characters, to access genetic data, and to access multiple sources of distributional data. Yeah, so maybe just one example from monographic work in taxonomy. We were, for example, of course, we were able by a huge and vast sampling worldwide from all available specimens from Indian fauna. We were not only able to extract and to describe a new species of those kind of chaver species, but we were also able to estimate species richness, but also then put this in a context of sampling density. So researchers actually can recognize how sampling density is correlated with species richness or vice versa. So that makes us clear that we can, from such monographic and museums collections worldwide, where we have areas to put more effort to investigate and in order to contrast our kind of biases. And but also with currently available georeferenced ecological and botanical data, we can, for example, then correlate or species distribution to certain biomes, and we can extract much more from our taxonomic work rather than just poor species IDs or whatever. We can really do biogeography and we can do ecological work, and kind of in the end, even if we don't have ecological observations and data collected directly associated with the specimen, we can extract from available global data, we can extract for the species ecological information. Of course, when we kind of taking data from collections into account, this cannot be just from one collection, like shown here for specimens of Ziricini chivas from Southern Africa. If we just take to account specimens preserved in our museum, which are marked in black, they are just a fragmentary information. But if we use more museums, I would not say all, but I mean, a quite big bunch of them, then we can get more or less closed surface data, much more continuous data, which are then allowing much better possibilities of niche modeling or to infer distribution areas or whatever kind of data. Of course, such kind of analysis you cannot do alone. You need involve people, and you must kind of transform your analysis in a way that also untrained taxonomists or students can help in processing data. And more of that, you can also use such distribution data in the end to establish, when you have also genetic data together, you can establish habitat connectivity based on your collection samples, and you can put this in confrontation also with, for example, patterns of land use or fire frequency. For example, we kind of use this in order to estimate the potential coverage with forest in South Africa, where forestry has led to a transformation of former forest into huge amounts of grasslands, which of course transformed already, in the past centuries. So nowadays there's almost no evidence for natural habitats, and there's a strong debate what's kind of the natural potential vegetation for certain mountain areas in South Africa. So we could use distributional data and niche modeling of potential forest species in order to reconstruct forest coverage. And so taxonomy can provide a lot of helpful tools in order also to assist conservation management. For example, also when we go in the collections now in Germany, we can very quite precisely reconstruct regional faunas by extracting the collection data we have, and that makes more, again, visible how necessary it is to have a continuous activity in collecting and monitoring faunas, and not only to make one survey and after that's finished. But for example, in Germany and Europe, we have traditions of continuous collecting over hundreds of years, at least the past 100 years. And we can reconstruct based on those data, for example, the development, disappearance or appearance of species. We can determine invasive species, we can determine outdying species, and we can also distinguish these patterns for different faunal elements. Like, for example, in Central Europe, we have an increase of Southern European elements and decrease of more cold-loving Northern European elements. And this is particularly interesting and important also, especially in the background of the general insect decline all over the world observed, and where agriculture also plays a very crucial role. So having good collections, building up collections, collecting every day in every area is also important and enhances then integrative taxonomy and the use of taxonomy, because we have the biodiversity inventories in time and space. Yeah, and then comes also new perspective for integrative taxonomy from other sources than taxonomy, which is kind of the development of artificial intelligence. The new sequencing methods in the genomics area, like methods like metabarcoding and genome sequencing, but also like already introduced initially, iterative procedures of integrative taxonomy, like the software IBBP, where we can simultaneously analyze molecular and morphological data, or even new possibilities in databases and networking, where we can easily extract from online databases distributional records, climatic data, etc. And so, yeah, let's come to artificial intelligence. So people have developed recently approaches of automated species recognition, either based on images, which is possible for all kinds of organisms, or even just for some, which using voices, like from birds, whales, crickets, and so on. And databases on these and the methods are developing really rapidly. Here you can see, for example, databases where image data are uploaded in the internet and where they have geographical references, done also often by citizen scientists. So you can see there's a huge community answering to this kind of field of researches, and the initiatives involving citizen scientists in Europe are greatly increased in the recent years. But of course, every kind of this new developments like barcoding, DNA taxonomy, or also automated image recognition needs the guidance of taxonomists, and even those very computerized and modern methods have their problems. And this is inherent in the nature of species and the nature of ecological habitats. This is a problem of the rare species and the problem of undersampling. Probably everybody of you knows that there are very rare species known only from the holotype, or if you make ecological monitoring, then you have those species which are occurring in hundreds of thousands of specimens, and then there are those species which are represented only by one or two specimens. And this kind of undersampling also impacts on training sets for those computer-based automated recognition methods. And so, yeah, we have, for example, explored or tried to explore what can be done in order to increase the accuracy when, for example, taking specimens and artificially increasing the number of specimens so the training gets better. And in fact, we could really show that the accuracy can be improved when we, based on training data with computer methods and different methods of increasing fake images, we can actually increase the outcome of those automated data analysis. But also we were able for the first time to extract a kind of like morphometric-like data from pixel-based images, so that we can use those kind of artificial intelligence approaches in order to automatically extract morphological features for integrative analysis with other traits, for example, DNA. So yeah, there are many ways of putting things together, but in the end, in all of this, the proper understanding of the methods and the empirical tests are only possible if you are a taxonomist who knows his species. So I already mentioned this, when we have this kind of iterative approaches where we simultaneously analyze different sources of evidence for species, like with the software IBVP, which is based on the multi-species score ascent in the analysis of multiple gene sequences. But nowadays you can implement also morphometric data, and you can analyze and then also select the choice of different traits, like using just linear measurements or nuclear loci, or using all traits like morphometric shapes or distance measures and all genetic loci and so on. So you can modulate your data and see what kind of outcome there is. And all this is the major thing is everything is reproducible. So any kind or anybody can redo again the same analysis with the same amount of data and can check why this specialist got this result in species identification or delimitation. And this is one of the major achievements or the major goals also to implement in taxonomy, that we can make testable our species hypothesis. But yeah, I already mentioned also new sequencing technology can come up with new sources of data, but so far one of the major problems with genomic data was that often the data used, like red sequence data or ribosomal DNA, were either not informative enough, or they were not applicable for all kinds of different organisms. And we have now recently kind of found an approach using universal single copy orthologs, which are represented in all kind of draft genome data, in transcriptomic data. And these data are, in the recent years, are produced for more and more less cost and for more and more study cases. So we found out that we can use kind of such a hierarchical framework in order to, using nuclear genomic data, in order to identify metazoan species. So the good thing is that it's applicable not only to a single group or a certain small group, but we can, in a hierarchical framework, apply that for a huge bunch of different taxa. And we have run an empirical analysis on different metazoans, including vertebrates and arthropods, where we could recover, for example, such orthologs for all 9 studied data sets. For more, we could recognize among them more than 300 different orthologs, which are kind of corresponding to genes. And so this could have kind of shown us that there is possible approach having universal and standardized data for a big frame of organisms and to having a large amount of data. And yeah, this would be maybe the kind of output we get. And this is the black one trees. We have, for example, now a tree what is based on those kind of USCO ortholog data, where we can see clearly distinguish the morphospecies evidenced here by different symbols. But this is compared with the same analysis based on CO1 data. And we can see CO1 does not is not able to separate all different morphospecies we used and which we could recognize between the different, using different USCO data. So the USCO worked much better, resolving the species much better than CO1 data. And we were able to align these kind of nuclear markers all together in a global analysis where we also could achieve very good results in higher level classification where all genera were identified as monophyletic, but also still the species, and we could recognize the different groups. And even using other sources of evidence like multidimensional plots on extracted from the SNPs from those USCO data, or doing structure analysis, we could very well recognize those data and those species much better than CO1 data. So yeah, I mean, and I already mentioned, of course, collecting and taxonomy isn't a very, very, very important crucial part in integrative taxonomy, because all of the methods I introduced and showed now, all are not possible if we have only single specimens. So we need multiple specimens for species delineation, either using morphometric data or using molecular data or genomic data, and especially also if we are using artificial intelligence. So that means that we must ensure that specimens are collected in sufficient number. And I have, for example, very often had this kind of problem that conservation management authorities allowed us to sample only a single specimen or few specimen from a certain locality or at all. So when we, for example, using molecular DNA taxonomy approaches for doing ecological surveys, we are not able to identify the samples because we are not allowed to collect the sufficient number of specimens. And the same is when we're having our museums collections. In the former times, specimens were collected by chance. Now we have to apply for permits and have to respond very strict rules of which of course they are for conserve biodiversity, but actually in their proper implementation, they are kind of a burden for collecting and for easily collecting without bureaucracy, and also which are having the impact that taxonomy community is declining. So these are all problems we have to face, and we cannot forget them when facing integrative taxonomy. And yeah, I have just in the end maybe of my talk, and I tried to sum up this a little bit also to give a kind of good time frame for all this. I summarized a little bit the outcome and the different observations. What I can take off from the talk is, yeah, we should always, in order to establish and re-establish taxonomy as science, and as you have mentioned, in India have the same problems of funding for taxonomy and yeah, all the different kinds of research related to taxonomy. This kind of problems are of course there because taxonomy was neglected to be a real science, because the people thought we are just describing lookalikes from species. And that's why we should use or try to use quantitative data, what can be realized, and to develop approaches that integrate different lines of evidence. We should use multi-marker based genomic data, and of course, even sequencing genomes became more and more economic. So in future, we expect that instead of using CO1 as a marker, we will use whole genome data for doing that. And we should seek to fasten up the data gain, to automatize, to develop workflows also that can include unskilled people. And this is what is one of the major, maybe thing normal, traditionally working taxonomists can do. We of course need to critically discuss all results, even if we use fancy molecular methods, because the outcome must not really reflect species boundaries or correct identifications. And we should also allow and test alternative and competing hypotheses. And all of you should start to think not to believe in the dogmas like barcoding gaps or so on. And I myself, coming from the background as taxonomist and applying the different methods of modern species delineation and even using genomic and DNA approaches, morphometrics, and so on, always think back to the roots and look at your species and as a specimen. And of course, there is this kind of competition, man versus machine, that we think we can better resolve with computers some problems, but there are currently many problems and failures of automatic approaches, and this kind of lots of subjective bias, what we find in kind of all of applications of what we know from modern taxonomic approaches like DNA barcoding, artificial intelligence. That kind of a priori subjective prior choices, even if they start from sampling issues, of course, rehabilitate in kind of traditional taxonomic procedures, which are subjective decisions of specialists, which are, however, all based on a proper and extensive training with specimens and species. Yeah, so all in all, taxonomy needs various approaches according to what we need to achieve, whether we want to, mirror world's biodiversity, or we have difficult taxonomic problems to solve. But we need to develop speed, we need to develop accuracy, and we need the taxon coverage. So, and with these words, I thank you a lot for your attention. I'm very happy to be that you invited me and to bring my knowledge to my Indian colleagues. And of course, I'm also especially thankful to Dr. Kola Sridevi, with who I have a collaboration ongoing since several years. And of course, I'm happy to also answer questions that you have maybe.
Yes. Yes, Dirk. Thank you very much. Thank you so much for that exhaustive and informative talk on the integrative taxonomy, especially on the species delineation, the DNA-based species recognition and species delimitation. So we'll be happy to take you forward for further talks in due course of our programs. And we are highly thankful to you, Dirk, for accepting our invitation and being part of this program and delivering your talk. Now, shall we take up the questions?
Sorry?
Yeah. Questions.
Questions.
The
Okay.
queries.
I'm just
the queries.
trying to check
in Q&A session, we have from Dr. D.V. Sai Ram Kumar. He was telling that please explain about us to how to analyze the insect samples of color morphs or ecomorphs, wherein the isolation is slight or incomplete, wherein there will be some differentiation in their genetic constitution.
I mean, that depends a lot on the whole framework. I mean, there are many situations where color morphs, for example, we know that can depend on the feeding of the larval stages on different ecological or geological grounds, that they have a different ion intake. So that can cause different color versions. But of course, when you check those different versions using first, for example, using barcodes, but even you can use nuclear genetic data, you can see whether there are distinct morphs, forms in terms of different species, forms, subspecies, whatever, or if they are part of the multivariate or ecomorphs or color morphs, which are representing not really evolutionary entities. So this would be the way of looking at this.
Yeah, I hope they got the answer. And also there is one more query from Saranya Gopalakrishnan that she is asking which method is currently used for data analysis of taxonomy. Like when we go for the data analysis of taxonomy?
I mean, this depends. I mean, I already tried to mention, I mean, for the most taxa, we have available morphological data. So still morphology is a method of choice in order to achieve those taxon coverages. And then also, I mean, nowadays we say, okay, barcoding is maybe prone of errors, but of course it is valid in order to identify many species and also to investigate environmental samples. So barcoding has still its use. However, it is like similar to a specimen image. It has a certain amount of information that can allow you up to a certain degree of correct information and identification, but it can be prone of error. So if you use multiple nuclear genes, this would be probably the best method and with the strongest evidence. So this would be the way to proceed. But of course, then the amount of reference data is much less. And for this reference data, we still have to work on. And so it will be a process and time in order to achieve, like in barcoding, that you have to build up those libraries, what you can compare with. But until even barcoding libraries will be complete worldwide for all species, we have to use morphology in order to be able to say whether a species is new to science or not.
Yeah. Yeah, that's good explanation, I feel. Then the another query from Satyan, he's asking whether can be explained about Bayesian index node. Is it based on the posterior probability? He's asking some question like that. Satyan, can you please explain
Okay.
us about
yeah. So I mean, we have different gene trees, like a certain number, and in those nodes where those gene trees are agreeing in their topology, in these nodes you have a very high posterior probability because the species tree topology is supported by a high number of different gene trees. So the node what is supported by a higher number of gene trees have a high posterior probability. So in this way, a certain node is likely to be a good species split if he's supported by many species, by many trees. But if you have, for example, within the species, within one different population, there's not this kind of species lineage that follows up. And so the different gene trees do kind of a lot of different things. And in those nodes, you will have lower probabilities because nearly every gene tree, because of lacking selection and because the time is not enough, and the coalescence and so on, these processes, you will have different gene tree topologies. And those nodes then, the intraspecific nodes will then have a lower posterior probability. And in this way, nodes with low posterior probability will result as conspecific nodes. So that's more or less the way.
Yeah. Yeah. In continuation of that, he's also asking about your opinion about the phyllocode. With more genomic data, will it be best alternative for existing zoological nomenclature?
No, because I already mentioned, and especially every phylogenetic analysis shows this, that phylogenetic topology depends very much on the sampling of taxa we include in an analysis, and also the choice of characters we use. That can start from if we, for example, exclude the third codon position of the genes, protein coding genes, or if we partition the data in a certain mode, which phylogenetic and evolutionary model we apply for tree search. So there are a huge amount of different variables which can influence the topology of the tree, that any kind of difference could cause a different topology. And so this is not the proper framework in order to establish a stable nomenclature. So, and I think there was also recently in Cladistics a review article about that phylocode is dead. So nobody has to worry anymore about this, because I think this became clear, but it was not so clearly said in any of the papers because
Okay. That's a good clarification. Yeah. Thank you for that. And one more query from Meghana. She is asking how much percentage variation should be there to consider it as a new species from a known species? Whether percentage nucleotide variation differ from species to species based on their morphology?
Yeah, this is one of those questions where I said that depends on your group. You have to compare what makes sense. You should compare different morphospecies and what kind of distances there are between the different morphospecies. And then you can maybe assume with this kind of gap, there could be different species. But then, of course, I would not put my hand anymore into fire for this, because we know, for example, that there are species like, for example, even tigers or lions, where the females or males don't disperse that actively. Especially in CO1, there are patterns of distinct clusters which can be more greater than we expect by the dispersal capacity of an animal. So, for example, in bats, it's been shown that they are very territorial, and there can be very deep splits between populations which are not corresponding with then splits what we know from other species. So divergence, especially in mitochondrial DNA, can be very easily, for example, also caused by a past range fragmentation, which is now again fused together. Like, for example, in rose chivas in Europe, we have Pleistocene refugia, which after the Pleistocene were reunited again, and we have molecular distances between CO1 and within one species of 5%. And for example, in the wingless species, what we have shown the example of sex-biased dispersal, we have between populations which are 10 kilometers separate, we have 5% of divergence. Between the species, we have 14 to 20% of divergence. So that's not corresponding what we have in the rest of insects, because yeah, when species are not dispersing very much, like myriapods or cave insects, wingless insects, they have much more intraspecific divergence than flying insects. So you must always look to your study case and verify this with external evidence.
Yeah, that explains clearly the group with which we works. We need to go by species wise or group wise. Then one more, Stuti, he is also seeking clarification on which analysis like NJ method or ML method or maximum parsimony to be used in analyzing the data, or at which place it can be used, she is asking.
So yeah, most of the DNA-based species delimitation methods use the information of number of substitutions. So that's why maximum parsimony methods are not so suitable, because we need to infer also the branch length or the number of substitutions between different branches. Okay. Once you have your genetic data, you can, I mean, there are several online portals and tools where you can upload your sequence data like a FASTA format. You can create a tree, and then you can take this tree and go to other servers and sites where you upload your tree, and then you get, like for example, with PTP, a species delimitation result for your species. So if you just Google species delimitation online, you can find maybe some of those, with the method name, you can find out some of those servers which are free to use and which are also kind of provide very quickly results.
Yeah, yes. Okay, thank you, Dirk. Thanks a lot for all your clarifications and also information and exhaustive talk. And we thank you for your presence here, because as you have messaged that you have another meeting to attend, we are delighted to have you here, and we are privileged to hear to your talk. Thank you so much, and we look forward to your constant and continued support and encouragement to us in building up of our referral collection or identification, all the other collaborative works. Thank you, Dirk.
Yeah, thank you.
Thank you so much.
I'm happy to help anyone who needs. So
yes. Thank you.
Bye.
Yeah. See you. Bye.
See you.
Bye. We'll move on to the next speaker, Dr. Peter Sipek. Who is working as assistant professor in Faculty of Science, Charles University, Prague, Czech Republic. He has had his education and also employment throughout in the Department of Zoology, Faculty of Science, Charles University. And his main research interests are taxonomy, evolution and ecology of Pleurostichtes scarab beetles, insect communities and heterotrophic succession on carrions of small vertebrates, and also insect communities in production hay meadows. And he is also interested in changes in insect biomass production and diversity. So he has several international stays and also academic appointments as member of the Academic Senate, Faculty of Science, Charles University in Prague, and also board member of the Czech Biological Olympiad. And he has research grants, many for his projects, and he is a principal investigator for 5 projects from the Charles University Grant Agency, and also project supervisor for 3 projects from the Charles University, and also EU Synthesis Grant Agency. He is into active teaching and also research, and he is supervising the undergraduate and also PhD thesis and reviewer for many journals, and he has publications of around 50 papers in journals of international repute. And he's specialized in immature beetles, and he's instrumental in starting the immature beetle meeting, and also active in going ahead in conducting this immature beetle meeting biennial at Prague in Charles University. And I'm very privileged and honored to introduce you, Peter Sipek, to this audience. And we have, as I said, we have a mixture of associate professors and senior scientist level, and also the students, scholars in this group. And we would like to invite you to deliver your talk, Dr. Peter.
Okay, Bahu Dhanyavad Kola. Thank you very much, Dr. Sridevi. I'm pleased to give a talk, despite I'm a bit nervous. I'm after Dirk Arendt, who is really one of the leading persons in taxonomy, or in the new approaches of taxonomy. And I guess he is the person who inspires a lot of other works. And so it's my big honor to speak to you and to all of the audience in India. Well, I would start. So I try to share my presentation. Okay, and start. Oops. Okay. Well, as introduced, I started my scientific works mainly due to my love to insects, but I loved the most living ones. So I tried a lot of breeding, and I did a lot of breeding of several species, of several groups of insects when I was young, and this led me to study entomology, because I was very fascinated by the development of insects, by all these developmental changes which are happening, and by this basic miracle which we see every day when we are outside in the nature. So this led me to study immature insects. Well, first, let me to start with this citation of Charles Darwin, which is not historically known, but still every day when I came across the diversity, I have to remember it. So, like, from a simple beginning, and endless forms, most beautiful and most wonderful, have been and are being evolved. I mean, these words are very clear and very, very wise. And also in the prospect of modern taxonomy, this is great that already Charles Darwin knew that the species are still evolved, and they are still evolving, which especially is complicating our situations, because nobody knows at which stage of development of new species, of separation of new species, or new lineages we are looking at. But still, we have to remember that the insects are quite numerous. There have been there are very various estimates about how many insect species we have. There are some more conservative, which are speaking about roughly 900,000 to 1.3 million species, which are already described in insects. And if you look to all other diversity we know, I'm speaking of diversity of multicellular organism or complex organism, we see that by far insects account for 2/3, at least 2/3 of the whole known biodiversity, and this is a huge portion. So actually we live in an era of insects, and the diversity of forms means also there is a diversity of roles, and these roles may have an impact on those or have an impact on the society, on the ecosystems and on the society of other other beings on planet, including humans. So well, if you would look, because I know most of you are agriculture entomologists, so we have to reduce a little bit the roles of insects to so-called ecosystem services, which are impacting ecosystems and also agroecosystems. So basically, if somebody asks you why are insects important, the basic question is the basic answer is they are providing ecosystem services. That means if you are speaking to somebody who is not fascinated by diversity but is more or less into economy or just general person, so you tell them they are providing ecosystem services, then he asks, okay, what are ecosystem services? So these are the notorious and very well-known things like pollination and predation, parasitism, or decomposing of organic matter. And then you can start to explain that that about 80% of vascular plants are pollinated by insects. And if you give a flower to somebody, that you give a result of coevolution between insects and plants, that the flower itself evolved due to the coevolution of insects with the plants. So it's great. And then you move on and you say, okay, there are lots of parasites and predators who control other insects or other animals, and this is also a very important role they play. And then you end by that, and okay, every organic or every organism on the earth produces some litter, either it's some organic litter, and this organic litter is decomposed also by many invertebrates, including insects. And you can speak about the well-known examples of dung beetles introducing to farmlands in Australia to reduce flies densities and etc., etc. But what is clear that most of these services which are provided by insects are overlooked until they disappear. We simply do not realize that they are done by the insects, that we need them until they disappear. There are several known or many known examples of such services or of other roles where insects serve as creators of diversity, as so-called ecosystem engineers. So I would like to mention, for example, bark beetle, who in eyes of the most people in Central Europe is now the destructor of forest. But if you look on the biology or ecology of temperate forests, spruce forests, in fact, periodical outbreaks and dying of the old trees in the canopy is something which triggers new diversity in the forest. It triggers it renews the cycle, and it's probably the way how these forests have existed for many centuries or for ages. And now when the bark beetle has reached our tree plantations, we are very scared about the die of the trees. But maybe it was us who did something wrong and let him to enter to this to this open plate. And of course, these ties and these things are very, very complex. And sometimes you see that that even a presence of a invasive butterfly can have impact on the number of people who are infected by Lyme disease transmitted by ticks, as was shown in this famous example already some 25 years ago in North America. So the ecological nets are very complex, and it is no surprise that in the end, it's no surprise that when they got broken, we have a reaction which we cannot expect, and we are surprised of. This all is in the prospect, and this all comes into the prospect with the recent findings of global biodiversity crisis in especially in insects, where we see worldwide decline of biomass and species richness of insects, well documented in several new papers which have been published in the last decade, and which shows us that especially the densities of the common species are quite frequently declining, and this means also we lose their ecosystem services, and we open up new niches for new species to come up, or for species which have been regulated by the former. But let's come back to the topic of my talk, because I was asked to speak about the immature insects. So what are immature insects? What we understand if somebody says immature insects. So these are roughly, if you say, all developmental stages before sexual maturity. Terms like egg, pronymph, nymph, larva, pupa, prepupa, or whatever, these all show or target these stages, which are part of the life cycle in a species. Unfortunately, most of connections and most in most museums and in all studies, we are mostly dealing with adults. However, we shall not forget that there is that the adult is only a part of the life cycle, and that an insect can have only a very short period as an adult, but very long as a larva or nymph or other pre-immature stages. So we have always to bear in the mind that if we look for ecological impact, we have to think about their ontogeny. So if you look on the development of insects, we basically have three groups, or three basic lines. One, the very plesiomorphic, is ametabolous development, where you observe that from the beginning till the maturity, the species have the same morphology, and that they are able to mold also when they become adult. These are this development is known for the Idli lineages of hexapod, for the Entognathous lineages, as well as for the Archaeognatha and Zygentoma, the two orders which are primary apterous. So in these orders, you encounter molting or growth after they become adult, which is much similar to many of their crustaceans relative from the crustacea, so-called crustacea. However, the derived development the more derived two types of development are called hemimetaboly and holometaboly, which occur both in pterygotes, in winged insects. Basically, hemimetabolous development means that there is no pupa in the development. You have a nymph, larva or nymphal instar, which is looking similarly to the adult, and from a certain point you can realize that it has wing buds and which are during the development growing, and the larva is basically resembling the adult specimen, but there is some exceptions in aquatic insects. Then we have the most derived holometabolous development, in which the larva is very, very different from the adult, and has very different feeding strategies and ecological strategies, and the ontogeny ends with a pupal instar, which is an inactive instar, which does not take food and is not moving. So, well, what are the differences when I sum it up? So the nymph in hemimetabolous insects, I put hemimetabolo in parentheses because it's paraphyletic group, so we don't really look on it like a group, a monophyletic group. The wings here are exposed, growing during molt. They are therefore also called exopterygota, while in holometabola, which is one clade, internal clade of insects of the pterygota lineages, they are internalized in structures which are called the imaginal discs. These discs are hidden inside the larva, and only and they are gradually growing, but not very well during the larval molts, but they become to grow rapidly or to develop rapidly between the last instar molt and the pupal molt. So then the other differences are that immatures of hemimetabolous insects, they have already adult eyes. Their eyes are already compound. They have dorsal and lateral ocelli, so they have fully developed sight systems. While the larvae of hemimetabolous insects, they have eyes, simple larval eyes, which are called stemata or larval ocelli. And of course, there are differences in their feeding habits and in their ecology, leading to differences in their mouth parts and in their general morphology. And this leads to a conclusion that the selective advantage of a complete metamorphosis of holometaboly is that within one species, you get two distinct forms with different lifestyles, life strategies, and which exploits diverse habitats. This has led to decreasing of intragroup competition, and allowed insects to explode or to get much more species rich than any other group on the earth. So before I get more into the perils of studying immature species, I have to raise this question. We still don't know, and we still don't understand the proper way or the proper difference in the between the development of between the hemimetabolous and between the holometabolous development. We still don't know where from pupa has arrived. There are basically two competing theories which have started, which have postulated many decades ago, but still they are not sufficiently explained, and there are competing evidences for both of them, even with the use of the modernmost Evo-Devo approaches. So the first theory is the Hinton theory, which it says that Holometabolous pupa evolved as an analog of or homologous to the last instar stage of hemimetabolous nymph. Basically, that the last nymph has become inactive and has become is modified to pupa, which means that all other larval stages are homologous to nymphs and larvae, and nymphs are homologous. Against the competing hypothesis is the Bedleze theory, which says that larval stages are something which is related to externalized embryonal stages or pronymphal stages. Basically, and it states or it sees that a lot of embryonic changes which are already finished once the hemimetambolan nymph is for the first instar hatches out of the pronymphal stage or egg already. This development is retained and is stopped for a long time and is continued in the last mold between pupa and the last larva and pupa. So basically, that a lot of embryonic changes and development happens in the pupal stages or between the last two stages. This would mean that the pupa is homologous to all nymphal stages as shown here. Well, it's the question is still the question. The debate is going on. There was the whole issue last year or 2000 or 2 years ago of the philosophical transaction of the Royal Prosody B dealing with these issues. So you can read more on this topic. But I wanted I thought it's important to mention it here because if you look on hemimetabolous insects, the larvae or the nymph, to be accurate, they're mostly exploiting the same habitat. They're mostly living on the same food plant or catching the same prey or similar prey, just in different size. But in Holometabolous, they are not the most numerous species. All like 83% or 4/5 of all insect species known are belonging to Holometabola, which means that almost 2/5 of species have two life strategies, and we shall not forget about it, and we shall also ask what is the ecological and economical significance of these immature stages. So some examples, well-known examples are grubs of white grubs, larvae of pleurostick scarabs, as already Dr. Sridevi mentioned, which are pests, serious agricultural pests. And here, as it's a quite a nice example, here we often don't see the damage when it's actually happening. And we don't see that their role until they hatch and there's a huge die-off of grasses and the feeding substrate. So the ecological roles of, and similarly to that, we can state that ecological roles of many immature insects are simply overseen. We simply don't know too much about them. Maybe we should also change our point of view and try to bear in mind that instead of this picture, we can also have this picture. Well, this is one of the point of the talk. So why to do studies on immature insects? We need them because many of them are, as I said already, are pests and can have a huge impact on our ecosystem and agro ecosystems. While the others can help us in the same way. They're predators and parasites or parasitoids of other species, and of course, getting information on image stages can help us in more in understanding phylogeny of groups. Of course, mostly this is done by molecular means, but however, the larval characters are more conservative, are not driven by sexual selection, and thus they can retain deep splits in the phylogeny and can be informative or provide morphological apomorphies for groups for trees identified by molecular means. And of course, it's also good to just complete the information on life cycle and bionomy. And it's always nice when you have a species which is less known and you know them only from few exemplars in the collections, then to be able to complete the information on life cycle and etc. And not to forget, in some groups, the identification, the morphological identification of immatures helps to separate species. It's especially in those groups where the adults are very, very similar. Many plant-feeding groups, e.g., larvae of Lepidoptera, caterpillars, or larvae of sawflies, they are distinct as larval stages, but very similar when they are adults. In general, immature taxonomy suffers from overlooking. If you get most images from insects collections, from museums, you will see only images of adults, of the beautiful specimens arranged, and it's because the adults are of course these target groups or these first groups which we see before when we start, and which we encounter in the forest and can be to some extent at least identified to genus level or to family level quite easily based on just how they look like, which is not the case of larvae. There are estimates that only about 2% of known species of Coleoptera larvae are known. This was 1990 estimate, but since then the situation has not really improved. And when we compare the number of species described here and number of larvae described here or immature stages, this is quite different. Okay, there are several faunas where this or areas where this knowledge is better. For example, in Central Europe, we have a huge number of families in which we know at least something about the larval morphology. Mostly, it's related to a work of some people. For example, in Cerambicidae, it's related to a lot of to work to Peter Schwacha, who also had some complications while collecting in India, so it's known from this case. And also in a recent time, new studies are developed. For example, this is some of the work done by Dirk Arends, the fourth speaker, And some studies related to agriculture, agriculture places, mostly done now in South America by South American researchers, or this study from Madagascar, or etc. So what are the challenges and perils? These are mostly widely you don't simply know the life cycle. You don't know, you know when how to catch the adult, but you know how to find the larvae. For this Bombelasmus beetle as an example, it's a great thing because it's very sought after by collectors, and many people would love to find it, but its larval feeding strategy is almost unknown, and people, and we have only some notices that they may feed on subterrestrial fungi, the larvae, but nobody has ever seen the larvae of that. Of course, I said the larvae are not the target of most collectors, and this is also because they are hard to collect. They are soft-bodied. They are their body is not sclerotized, and they have to be collected in a separate way. Mostly, they have to be already in situ killed by boiling in water, and then emerged either in ethanol. But for several groups, you need to use different solutions like Pemple solution or card solution, both containing various chemicals like kerosene or formaldehyde or acetic acid, and they are not really pleasing to work with. Some of them are really also toxic, and also the storage of the larval material in immatures is much different from the storage of adult material. You have to have store it in alcohol in some closed vials, which are which serve as environment for small vials, and these are arranged in some boxes which should be climatized to prevent the evaporation of alcohol. And this kind of collection is a very time demanding and space demanding. However, this is, for example, one of the biggest collection of larvae which is housed in Smithsonian, and you see this is only one single room which houses almost all larvae of Orthoptera. So, and if you compare it with the amount of the adult material collected, it's really different. Then you can come up with all the old collections have problems like the preservation mediums get dry out, so you need to rehydrate this material with some solutions. Thanks God it's possible, so we've been able to rescue this larva, which is one of the 4 specimens of this African trichine species, which is pest species to or used to be pest species to oil palms in equatorial Africa. So we've been able to rehydrate it. And another really challenges or challenging things are that there are different to describe or to work with the larval taxonomy is quite demanding in terms of visualizations. You simply don't go along only with the image of adult. You have to dissect the species, and you have to make the descriptions much more complete. So for one species, you use up to 15 structures which need to be depicted, at least in my group, or at least in the larval characters. So this makes the work with larval taxonomy much more complex than one can say. This is an example from rose chaffer families, from genus Orthoptera, from one of our papers. Then you see this is a huge amount of work which has to be done if you want to properly document the morphology of the species of the immature stages. And another or the other in the other way, or the other problems which are is that these other challenges is that if you go in a field you collect larvae, how can you tell that the larva is belonging to some particular species? How can you associate it to a proper species? So in the past time, frequently the larvae have been attributed to the habitat of adults, simply meaning, okay, the adults are living here, the immature stages maybe are also found here. But especially in holometabolous insect, this is very tricky, and this is not easy. Sometimes it's easier if you find a stuck beetle and larva in the similar wood. It may be the same, but or here says the example of Porphyronota. But generally, it's not the easy way, and it leads to many misidentifications. Much more precise is an association with DNA-based methods, when you have a reference sequence of an adult and you match it to the larval DNA larval sequence. However, as already was shown by Dr. Ahrens, there are also huge pitfalls which can happen, especially if you have a multi-species assemblage, or if your sequence of your larva is not originates in different places from where the adults come, and there can be big divergence in the mitochondrial DNA. So also these techniques are quite, we have to be cautious while we use them, but still they represent a good tool to work with. Another possibility is to work with to identify the species based on flow cytometry. It's used, for example, in approaches in forensic entomology, etc. This gives us. We have also to have a reference value from adult. This method is based on the knowledge of the C value, which basically translates as a amount of nuclear DNA present in cells. So, but here the biggest problem is you have to work with live preserved animals. So basically you can match adult and larva based on the amount of DNA which is found in the nucleus. However, there are also groups where the differences at species levels are not really that high to compete with intraspecific variability. And the most precise way, however, the most time-consuming, is to perform self-breeding of the animals which you have in your which you collect in the nature. But this is possible only in some groups where the animals can be bred, or you have to exhibit to be very patient, and you have to work very precisely, and to be able to work with living stuff. But once you can associate the adults with the larvae which have been produced by them, you get the best idea about what is the about the identity of the species. So before I end, this is only a basic introduction how to work. There are another pitfalls when you are working with immature stages, and these are that these species have different developmental stage, and especially in some groups, it's quite important, or it's very important to know the instar, the stage, the development, the correct larval stage which we have in your in the hand. So it's quite hard to say if it is first instar, second instar, third instar. And for some types of works, identification or phylogenetic works in, for example, carabids or other beetles, it's important to know it very well, because the instars are very, or not very, but can differ among themselves. So if you use, for example, in carabid beetles, the determination by primary chetotaxy, you have to know that in second instar and in third instars, you get different patterns of chetotaxy and setae on them. So this is another complication, and often because the size of the larva depends in holometabolous insects very frequently on the size, on the feeds of on the source of food, on the substrate where it's developing, a number of food consumed, you get also big overlaps between in sizes between the various instars. So this was one of the original of the idea, is to count to tell the instars based on the count on the size of head capsules, for example, or on the ratio of the size values or metric values in this. But you can, especially if you breed the specimens artificially, you can get different results based on the conditions you rear them. So to sum up, I have to state that the larvae are quite understudied, or the larvae, especially in holometabolous insects, that 2/5 or 80% of insects have two different life strategies, and that the larval strategies are often overseen and neglected, and often also in the stage that the immature stages are indeed those more economical important, and that larva stages are or do have or are informative for the reconstruction of phylogeny. As Dr. Sridevi already said in the introduction, we also built up a small meeting for coleopterists who are working with immature stages. So you can see these are our flyers. The next one is scheduled for this December, and we will this November 2021, and we will see how all the things will happen. So we will post some news about how this immature beetles meeting will be held in the current situation. So thank you for your attention, and if there are some questions, I'm happy to announce to reply.
Yeah, thank you, Peter. Thank you. That was an excellent talk, elaborating the procedures and also the details of how to go about the immature stages taxonomy, and what are the pitfalls and what are the challenges, and how we should proceed to go with the immature taxonomy or the characters diagnostics. Thank you very much for enlightening us in the basic aspects, and also how difficult it is to go for the immature taxonomy, and how important it is to go for also. Thank you so much, Peter. We just see if any queries are there.
I have one question, Doctor.
Yes.
The cuticular hydrocarbons based identification, which we have been discussing, but never happened. What is the opinion of Dr. Peter about that?
Well, yeah, the method is. I've heard that people were discussing it and they were trying to use it, but actually this is not really happening, and mostly also because they can vary. The cuticle hydrocarbons can vary also among places in for example, social insects. They are very variable according to the nest of the, for example, in ants, the different nests have different composition of hydrocarbons, and they are very difficult to collect samples which retains these cuticle hydrocarbons unchanged. So I don't really think this is a promising method, and this is also very costly, and you don't get any. If you do sequencing, then you have the sequence and you can work with the sequence. But if you have cuticle hydrocarbons, you know the composition of cuticle hydrocarbons, and this is it. You cannot use it for, or it's quite hard to use it for any other application.
Of course, when I was studying for the Chiromones effect on for this larvae on the parasitoids, we have done so much of analysis, but we never thought in that angle. Let me see if I can have some collaboration with you, and we will try to develop some kind of database based on that results which we have got. Because I have an opinion that it can be a better thing if the protocols are properly developed. So protocols are very important. So the protocols are properly developed. I think it still has some hope on that.
I mean, in the way of you are speaking that of the application you are mentioning, it has a huge. I mean, in the study of chiromones and semiochemicals, it is very important. But I would hardly say it's important, for example, for identification of the species or cross or matching adults to larvae. But to be honest, I'm not really skilled in this technique. So maybe you know more than me, or you know more than me. So you have more. But it would be exciting to test, if, for example, if there is differences between species, and if there are differences between variously bred species or species under different ecological circumstances.
So there are differences in the proportion of different hydrocarbons that we have seen. So maybe we didn't look into that in that angle. Maybe as you are working on the immature, we may have some, because the protocols, everything, if you want, we can help. Sridevi is there, so we can try to help it. So we will see something we can do it on that.
Okay.
Thank you, Dr. Peter.
Yeah. Thank you. Thanks for that.
Dr. Varghese is there. He's looking and he wants to.
Yes, sir. Varghese, sir.
In fact, I was only enjoying his lecture. I don't have any questions. I have only to make a remark. It's quite challenging to take up immature insect taxonomy. You know, in fact, when we were in college, when we were asked to take that course, we ran away from the professor because it's such a tough thing. You know, so you taking to it is a remarkable thing. My having Bhaktavasalam having prompted me to make some comments, I just want to know whether the ecology per se, you know, the kind of hosts that, you know, say a polyphagous insect like Helicoverpa, which has more than 200 hosts, and the immature insects in that, you know, would it, what is the challenge? Is there a challenge for a polyphagy and a monophagy immature scenario? Is it easy, or it doesn't make any difference?
No, it's in fact it's quite complicating the situations, because we know many lineages in Lepidoptera which are not, for example, in Lepidoptera in the plant feeding groups, which are really, there are lineages which are strongly adapted to their host, but they're still the same species, and or they're still believed to be the same species according to virus markers. We don't know in the stage of the species evolution where we stand, but this is quite. I mean, this is a very important, for example, in parasitic species, in species which are bound to a concrete host, but it's less specific in it is less specific in saprophagous species. But on the other hand, there are strong preferences to special types of substrate. We know that dung beetles, they are not taking all type of dung. Some of them are specialized for dung of ungulates. Others are specialized more for dung of primates. And there have been like also this example which I've been talking in the beginning, was that in Australia the local dung beetles were not able to take the dung produced by the cows, because it's totally of its totally different structure, and well, it's still dung. We would say, okay, it's dung used by kangaroo or by this species, but so it's taking an account, and also in many saprophagous species, we'll see there is strong preference for one or two or three types of substrate.
Okay, okay. Thank you for that reflection.
Yeah, thank you. We have one more query from Judith. She's asking that will there be any change in the larval period and also the activity while rearing them in laboratory condition than in the natural habitat?
Well, this is very good question. It's a good question. Basically, the larval period is somewhat, or what can I say from the place we are, so in the temperate zone, the larval period is something which is quite, which is hugely dependent on the temperatures, on the temperatures of the environment, and thus it's very plastic. And once you get stable conditions in your laboratory breeds, it's quite possible that either the development will not be completed because the larvae have their programs for having a diapause, for have a overwintering phase or phase of inactivity in hot times of year, when we are speaking, for example, in specimens from species from the Mediterranean, or you will get slightly different results or shorter development under higher temperatures, which also will correlate with survivorship. For example, we have learned that a lot of larvae which develop very fast, they have worse survivorship when they are adults, because their development was too quick, too fast, and they have used a lot of resources only for the development, not to be not to save some energy for the adult stage. So, well, it will alter the development, but it is the only way how to have the control about it is to rear it laboratory. So there's a strong, there are two competing points. You don't get the true natural conditions, but you get at least some information, which is important, because you start with nothing mostly.
Yeah, thank you. Thank you for that clarification, Peter. Any more questions from the panelists? Any queries or clarifications? Yeah, if
Thank you, Sayali.
there is nothing, yeah, yeah. Sayali from Pune, she was there, I think, in Charles University
Yes,
for
yes, yes,
some
I
time
know her.
ago. Yeah, yeah. So thank you so much, and very good to hear to your talk, and also for the information that you have disseminated. It will be very useful for the enthusiast who can take up the larval taxonomy. Thank you, Peter. Thank you so much. And in this junction, I would like to say that Dr. Dirk and Dr. Peter both have traveled India and Nepal and adjacent parts. And Peter can speak little Hindi. When I was there in Charles University. Thoda sa. Yeah. Yeah, that much is enough, and we are
Unfortunately, I
delighted.
forgot everything. So
Yeah, but still you could remember. It's a long time that you have visited India, but still we are happy that you can remember few words. Dhanyavad, thoda sa, like that. So thank you, Peter. Thank you so much for accepting our invitation and being part of it. And I would like to know whether you will be staying till plenary session. We have now Dr. Swaminathan's talk, and after that we'll have a few young researchers who can flag the issues of taxonomy, and then there will be a plenary session where we'll be taking all of your inputs. Our former directors are also there. We'll be taking their inputs too. So we will be delighted if you can, if your time permits, if you can stay back till plenary. We'll be happy.
okay, I'll let you know. Yeah. Okay.
Yes. Thank you,
Thank you.
Peter. Thank you so much.
Thank you for the invitation. I'm happy, and greeting all to India, and have a good time. And thank you for the invitation for this plenary for this conference.
Yes, thank you. We will now move on to the next speaker, Dr. Swaminathan. He is working as emeritus scientist at Rajasthan College of Agriculture, Maharana Pratap University of Agriculture and Technology, Udaipur, Rajasthan. And he is well known as or associated with the Orthoptera taxonomy, grasshopper taxonomy, and he is into the teaching and research, having 33 years of experience in MPUAT Rajasthan. He has guided several students in his illustrious career. 20 MSc students and 18 PhD students he has guided, and presently he is working as emeritus scientist in MPUAT, looking after the Orthoptera collection and also other collections at Rajasthan College of Agriculture. He has other additional assignments also when he is working in MPUAT as in charge placement cell, member academic council, honorary university librarian, and was PI for several projects, of which we are happy to say that he was the principal investigator for the ICR network project on insect biosystematics for nearly a decade, 2005 to 2017. And he was also for a short period dean for the Rajasthan College of Agriculture, and he has around 110 publications to his credit, and he has a lot of credentials, and we would be delighted. And it's a honor, Swaminathan sir, to introduce you to the audience. And we, as I have earlier emphasized, this is a mixed group of students and also scientists. You can, I welcome you to give your talk now. You can come forward to have your talk, Swaminathan sir.
Thank you, ma'am. I hope I'm audible.
Yes, sir. Yes, sir.
Yeah. First of all, let me thank the ICR and the organizers for inviting me and asking me to make a presentation. Particularly, the concern was of locusts because that belongs to Orthoptera, and really, I was also happy that I was involved with them, and I had been updated right from April 18 since the last, time the locusts had come in, April 19 up to, and I went up for a collection in February 20 also, and that was, I think, should have been the last swarm of the locusts that had come into Rajasthan. And right before my eyes, I could see that we were in a place some 280 kilometers from Jodhpur, and from there the swarm was migrating towards Pokhran. The scientists would not be able to tell them, but the local people, they were telling, sir, it is moving towards Pokhran now. Now it will go over there. And then it is going to next to Jaisalmer. From Pokhran to go to Jaisalmer. So they are very well versed. The local people are very well versed with this. And long back in 1999, I had been given an FAO student who to work only on locust. He was already working in locust organization at Yemen, South Yemen, and he had joined me for his MSc, and we took up the work at Bikaner. And 1 year we could do some work. We had reared the solitary group only. We had the solitary desert locusts, and we multiplied them into large numbers, then tested a few chemicals and wanted to study on the food habits, and also worked out some little work. Anyway, that was the only one chance that I could get. Well, then staying at Udaipur, and I'm highly thankful to Dr. Viratmat and Dr. Ramurthy, Dr. Verghese is here, Dr. Prashantji is there, who all helped me and guided me while working with the network project on insect biosystematics. Though I was new to taxonomy at that time, very much put into that. But probably Dr. Ramurthy was having great expectations from me that I would definitely come up. And well, we are into it, only into the traditional one, not into the molecular systematics so far. Well, I'll make my presentation. I'll just start my share. As I was requested to be more concerned about the locust, I will start with the Orthoptera diversity in the Indian subcontinent. And people often call many of the grasshoppers to be pests. So I wanted to emphasize on the pestiferous status so far that we know, and then come up to the locust. So this is the diversity. Just in a brief way, I wanted to show how much diverse the Orthoptera is, and more so in the southern part as well as in the northeastern part, the diversity is much, much more as compared to the other parts. Although we were restricted to Rajasthan, MP, and Gujarat, 3 states only. Though we did go for some collections, had the network project run along still more, it would have been even better. Anyway, well, in the Indian subcontinent, we have among the 2 groups, that is the Coelifera and Enchifera, 6 superfamilies in each, and 10 families in Coelifera and 11 families in Enchifera.
Sir, your slides are not visible.
Sir, slides are not shared, sir. It is still in sharing mode only.
Okay, I'll just once again.
Once again, you can start sharing, sir.
Yeah, I'll just stop share.
Yes, sir.
Is it visible now?
Yeah. Yes, sir. Yes, sir. You can make it full screen mode, sir.
Yeah, full screen
yes,
now.
sir. Yes, sir. Thank you, sir.
Okay, sorry for the trouble. Anyway, so I was talking about the diversity that is more in the northeast and in the southern states, and of course in some other places, whereas the place where we were concerned to work, it was a less diverse. The Indian subcontinent represents 6 superfamilies in Coelifera, and likewise 6 superfamilies in Enchifera also. But the number of families are little more in Enchifera as compared to the Coelifera. And we all know that they are shorthorn grasshoppers, and the Enchifera are the longhorn, including all these. Then as far as the Orthoptera species file is concerned, they have reported a valid species count of 28,840, which may have updated even today by there may be an increase. And there are a number of reports that only 10% of the accredited species are pest potential, are having serious pest potential. And the same way, Lockwood also has given this view that the vast majority of Orthoptera are not pests. Therefore, whatever management strategies we take up should be with great caution, so that we do not lose the useful ones. Then they have put it into 2 groups, the threatened species and the pestiferous species. The threatened species need conservation. They are usually large size, narrowly distributed, specialist feeding modes. They are monophagous or oligophagous and less mobile. Whereas the pestiferous species, they are smaller, widely distributed, polyphagous, and highly mobile. That's a very well known example of the locust, and particularly the hieroglyphus group of the grasshoppers. Then, coming to the mode of their feeding, phytophagy, omnivory, carnivory. In Enchifera, the Grylidae is known to have all the 3 types, whereas Grylotalpidae having phytophagous as well as carnivory mode, and the tetigonids both also having phytophagy and carnivory. And interestingly, Filifera is purely phytophagous, mostly phytophagous. No, I don't have any reference of any carnivory reported from the acridiids or the pyrgomorphids of the tetragids. Then phytophagy in Acridomorpha, when we talk of it, we have acridiidae the most important, and coming to that next is pyrgomorphids. Then locusts, earlier they were all in one, Pyrgomorphini is only one subfamily, but of late it has become a separate family. We have 10 global species of locusts, and of which 3 are in India. And among the grasshoppers, we have very few pestiferous groups, all of them in hieroglyphs. More than 8 species have been identified in this. And in Oxpia group, we have more than 21 species, but not all of them are very serious pests. And among the pyrgomorphids, we have Ollarchius, Colimania, Crotogonus, and Atractomorpha, although very few references to Protogonus and Atractomorpha as pests, but they are minor, mostly minor. Talking of the magnitude of pestiferous Orthoptera, it is very a difficult task. As Jago has pointed out, the global quantitative assessment is very difficult, and that can be done after prolonged infestation that has been accounted for. You have to record prolonged infestation record should be there, and then we can account for the global quantitative assessment. Most of the pestiferous species are in Acridiidae, and there are more threatened species in the less speciose family. Well, the reasons for their becoming pestiferous is long-term changes in farming practices, displacement of farming communities due to war or disturbances, which the recent locust upsurge is one of the examples of displacement of farming communities due to the war or disturbances due to the terrorist activities. Also, proper monitoring of locusts could not be done, and it had risen into a very much big upsurge this time. And poor crop monitoring is also one of the reasons why Orthoptera become, not only Orthoptera, to all other pests also it is applicable, but since I was supposed to emphasize more on Orthoptera, these are the reasons that they come. Then the centers of pest activity has been put up by Jago in 1998 with special reference to locusts. We have 4 centers: the North Central America, Northeast South America, and Southern Caribbean. Then Eastern Europe, Central Asia, India, Bangladesh, Afghanistan, parts of Arabian Peninsula. Then the Sahelian Africa, the African group entirely, Ethiopian highlands, from where we also get our group of the locusts from there. Then the China and Philippines and Australia. These are the 4 centers of pest activity of particularly Orthoptera and especially locusts. Then coming to carnivory, omnivory, and some reports that have been mentioned, like here this Caesodactyl is purely carnivorous. The Indotrella angusta is a grylid which is having mixed sort of a feeding habit, and the Hieroglyphus group which has got a purely phytophagous group. Then among Grylotalpidae, Grylotalpa genera as such are omnivorous and phytophagy and are pestiferous. Scapteriscus has been shown to be carnivory as well as phytophagy.
I will make one call to home.
Most Grylotalpids are omnivores. Then among the Grylids,
This one is also there.
Ecanthini, Nemobini, Trigonidae are predominant, whereas the others show omnivory and phytophagy. Likewise, in Tetigonidae, Listroclini are predaceous. Conocyphilini can be phytophagous as well as carnivorous. Mormon crickets are purely phytophagous, and most catidids show carnivory. This is the overall generalized view that I have taken up from various references. Then coming to the Indian species, we have Scapteriscus ecleatus as the carnivorous one, whereas the same Scapteriscus vixinus is herbivorous. Grylus assimilovorus feeds on grasshopper egg pods, so it is carnivorous. Pictonimobius is again a carnivorous one, feeds on the eggs of the velvet bean caterpillar. Metioke vitalicolis has been shown as the predatory on the eggs of various paddy insects. And Conocephalus longicornis has been shown to be predatory, whereas Conocephalus maculatus is carnivorous. And recently, I think in Karnataka, in the bamboo forest, there was a huge infestation by Conocephalus maculatus. Then in Listroclini, the entire family, subfamily is predaceous. Schizodactylus is again predaceous, and more than 100 species in grasshoppers belonging to Acridiidae and Pyrgomorphidae are herbivorous and mostly pests of the agriculture as well as the pasture. So wherever there are well-defined pasture lands, you find more of the Acridiid pests, whereas in agroecosystems, herbivorous group mostly confined to the hieroglyphus or the Oxia group, and in the Pyrgomorphids, the Colemania and the Olarchis group. So here some references, some status has been given in various references. I'll just present here. Grylotalpa and Tarbinscalis in paddy crop has been shown as minor. Lyogralis in pulse crops minor. Tarbinscalis in oilseeds has been minor. Again, Grylus and Tarbinscalis in cotton in June, again minor status. Then Grylotalpa and Tarbinscalis again in sugarcane has been shown as a minor. Grylotalpa in potato and Tarbinscalis in chili, minor to major. From Gujarat, we have received reports of Grylotalpa infesting potato crop and reaching major status in some places in certain climatic conditions. Then Tarbinscalis in Grylodes in citrus and mango in fruit crops as minor. And Schizodactylus is also shown in indigo and tobacco in the some of the condiments. It has been shown as a minor pest, but basically it is a predatory. Then these are some of the pictures of the predatory carnivorous ensiferans that we come across, the Hexacentrus and the Schizodactylus here. Then in Celifera, phytophagy is more common, and Hieroglyphus, Oxia and Locusta, and of course Cysticerca we can say when their locust swarms arrive, only then that at that time they are very serious. Otherwise, among the resident Acridiid species, we have the Hieroglyphus and Oxia, mostly in paddy and rice, Hieroglyphus and Parahieroglyphus in maize, sorghum, and most of the millets, and they become from minor to major. Their status have been recorded in different years. Then Atractomorpha, Crotogonus among the Pyrgomorphids, then Crotogonus, Colemania, Psittacanthacris again in Acridiidae. Although Psittacanthacris is called as a cotton grasshopper, but I doubt that it is a very serious pest. It's only a minor as far as it is concerned. Whereas the Pyrgomorphids like Colemania and Orthoptera can become major at many places, particularly in the south or in the northeastern parts. They are becoming major, some crops. Then again, this is in continuation. You can see Acridiids in cotton have been shown. Lastly, a few years back, couple of years back, we had Piculocirrus as a serious problem in cotton. And the reason behind that was, they had removed the weeds and removed the Calotropis growing wild. So once you remove the original host, naturally this insect, which is not a major pest of any crop, becomes a major. So when we remove it, human, I would say anthropological reasons are more to blame rather than the insect itself. Well, in sugarcane, Hieroglyphus banyan particularly and Oryzivorus group are more important, and Colemania has been shown as an important pest, but it's such a minor status. Likewise, in vegetable crops, most of the Pyrgomorphids and some of the Acridiids are yet under a minor status. Because it's achieving it to call a pest, when we follow the system of identifying it and then how to estimate the damage, we follow the two-plot design, the one controlled and the other one with treatments. Now to restrict only one insect to cause the damage, it becomes really difficult. You will have all the other insects also, not only the grasshoppers. So when we estimate damages, it becomes really difficult. So, so far, proper estimation is required to come up to the status. Then in the pestiferous genera coming ahead, again, continuing, we have among the fruits, Olarchis has been reported as minor and sporadic. Then Piculocirrus also, Ceratodus also has been, that is the hooded grasshopper. Ceratodus is also known in grapevine. And Orthoptera on cardamom. Then Hieroglyphus and Crotogonus etc. in dyes and drugs, minor to occasional. Then in plantation crops, minor to major, particularly last year itself, when the locust invasion took place, many people sent me photographs of Olarchis on, the coffee plantations on the palms and on banana trees, banana plantations, and confusing people, getting confused that whether it is locust. So that instigated me to that. I think proper identification is necessary so that people know where locusts are and where pyrgomorphids are. So it's different. Then in green manure plants also crotogenous and epichromia has been shown as epichromia are the acridids, whereas crotogenous in pyrgomorphids. They are having minor status only. In garden plants also they are occasional and minor. So as far as the pest status is concerned, only acrididae has importance as a pest in most of the cases, and to some extent pyrgomorphidae in some of the crops. Otherwise, Orthoptera as a whole, as it was shown that only few orthopterans, or 10% of the pest status they acquire. Then here some photographs see Gisonula on colocasia, because it is Gisonolia like Oxia. They like to be in the more aquatic habitats and they are found there. Then hieroglyphus species, here the Nigrolepidus and the Danian. You can see this is on sugarcane, this is on maize and sorghum. Then here the different species, Danian, the Oryzivorus, the Parahieroglyphus bilineatus, and the Nigrolepidus, these are the major common ones which are on sorghum, on many paddy crop, on maize particularly. Then here again, see cotton behind is safe, whereas the grass that has been eaten away. In maize, it causes a serious damage. Both Parahieroglyphus as well as Hieroglyphus causes extensive damage, particularly in Rajasthan, particularly in Gujarat, in some states, and in some parts of Maharashtra and Madhya Pradesh also. This is one entirely damaged in 2011. This was the status. 2011, 2007, 2005. I will show you the next slide. It is coming, how we have recorded it. And the entirely stripping of maize crop, 100% damage. The farmers had to face a serious difficulty because of this hieroglyphus group. It was not one species. It was having hieroglyphus Nigrolepidus, the major one, Parahieroglyphus. Then we had Oryzivorus. Then we had the other ones also in this. This is Banyan on sugarcane. Then Oxia group. This is the second most important acridid, which is more than having 21 species. And I'm happy to say that in Karnataka, people are working on it. And one particular student is in touch with me, with Kaiswaram Swami. She's working, and they are working on Oxia group. It's a difficult group. It's a really difficult group because there are so many similarities that you have to be very cautious before defining the species, coming to the species. Then there are some other ones which are minor, like the Coreodochus on banana. You can see here, Citracus, which is often confused with the desert locust. Then Echorifa and Itriponemus and Heteracris littoralis. These are some of the common acridids which are found in agroecosystems, which may cause some minor damage, not major. We cannot call them pests because they are food to the birds and to the other mammals and other animals and reptiles. So we have to live with them. Then see, this is what I wanted to emphasize on. Regular pestiferous occurrence in Rajasthan, MP and Gujarat in 2005, 2007, 2009 and 2011, every alternate year. And what happened was another alternate year, we had blister beetles. And we all know that the trianguline larvae feed on egg pods of the grasshoppers. They feed on the Orthoptera egg pods. So that could have been a link between the grasshoppers, Orthopteras coming, and the blister beetles, the what you call meloid blister beetles coming in the large numbers. So 2005, 7, 9, 11, and 2006, 8, 10, and 12, we had blister beetles. So that was the kind of activity going around. Then mass mortality we obtained because of the Entomoptera grylae attack in Baswada, in many of the parts of Rajasthan. And after that, we have not seen hieroglyphus infestation at a serious level. So we should say thanks God to the microbial infection that took place, and it contained the population. And another reason that I would like to emphasize is there was crossbreeding. We found male Nigrolepidus breeding with Cataloicus indicus females, and that could have also resulted into the sudden disappearance of this important pest in most of the maize growing areas here, maize and sorghum growing areas in Madhya Pradesh, Rajasthan, and Gujarat. Anyway, that could be, we could not work on it, but that is my understanding that we could have been one of the reasons, apart from the Entomoptera grylae infestation, infection that took place. Then among the pyrgomorphids, we have these common pests and Piculocirrus taking up sometimes pest status when it becomes serious, whereas Olarchis and Orthoptera are important. Then what impediments we do have in taxonomic impediments? Because the first one we can say is that only 15% of the described insect fauna we are knowing. We need more to know. And of course, in inventory and biodiversity is greatly hampered because of the taxonomists becoming rare. Then relatively it is benign for Orthoptera because Orthoptera is helped with the Orthoptera species file group, and we are able to have a good collection and good identification reports. And secondly, acridiids being of agricultural importance, also Orthoptera has been covered to a great extent, whereas the other groups of insects are not much covered, though in Caribbean also we are having the World Organization and most of that are being reported. But overall, in the last, I would like to say that major reinvestment and is relevant and essential with regard to taxonomy. That is one way we can overcome these impediments on taxonomic work. Then some more things that have to be taken into for the morphological taxonomy or the basic taxonomy before going into the biotechnological aspects. Use of common terminology is important. In some cases, common terminology is wanting. And then understanding the phenotypic variations, particularly in the tetrigid group, when we saw in tetigonids and in grasshoppers also, the phenotypic variation. One example I would like to cite very recently, like Oxia japonica and Oxia bolangensis. They look very similar. They are two different species, but they look very, very similar. The phenotypic variation has to be brought into consideration. Then study larger sample sizes. Then intraspecific color polymorphism should also be looked into. And for that, we have to have developed improved keys with drawings and photographs, and then verifying with molecular data. So all this has to be taken into account to upgrade or improve upon. Of course, many of us will agree to this. Many of the taxonomists do agree to that. But because of the poor funding in some cases, we are unable to do this most of the things. Then coming to the locus, the major topic which was to be told. See, a very famous saying that all locusts are grasshoppers, but all grasshoppers cannot be called locusts. To be called locusts, they have to exhibit two important things. They have to show density-dependent phenotypic plasticity. And of all the 10 known grasshoppers which are called as locusts, only 7 of them show the second character, that is the character of color polyphenism. So not all the 10 can be called even locusts. But anyway, world over, we recognize 10 grasshoppers as locusts, which are these which belong to Cyrtocanthracrididae. 5 of them are in Cyrtocanthracrididae. Oedoporini, we have 3. In Gomphocerini, we have 1. In Calyptemini, we have 1. These are the 10 species that are commonly known as locusts. Then we have the common names given to them. In Locusta migratoria, we have 2 subspecies, the Migratoria migratoides and the Migratoria orientalis, the migratory locust. Then we have the brown locust, the desert locust, the Central American locust, the South American locust, the red locust, the Moroccan locust, the Australian plague locust, the Bombay or Indian locust, and the Italian locust. These are the common locust species. Somehow the tree locust, that is the Anacridium species, which is, of course, maybe a locust, a pest problem in the African countries, but in India it is not to be called a locust because it never has that much of proportions to be called as locust. Here, the geographical area based upon the distribution, the desert locust is the widest, having the largest area under distribution from West Africa to Western India. The African migratory locust from Africa. In Africa, the oriental migratory locust in Southeast Asia, the brown locust in Southern Africa, the Central American locust in Central America, the South American locust in South America, the red locust in Eastern Africa, the Moroccan locust in Northwest Africa to Asia, and particularly Central Asia, the Australian plague locust restricted to Australia, the Bombay locust southwestern to Southeast Asia, the Italian locust in Western Europe to Central Asia, and the tree locust in Africa, Mediterranean regions, and the North, Near East particularly. The tree locust is considered as a serious locust problem. Then the desert locust, the lower picture, the yellow one, is the mature adult which we received in 2019 in May, and this is the immature adult locust female which I collected in my own house here at Udaipur, which was which was from the flying swarm. It had come down, and this was collected here in a basket. So you can see the color differences in the immature adult and the mature adult. So the desert locust, coming to the phylogenetic position of this desert locust, it's the only old world member and the earliest of all the diverging lineage within the genus Cystocerca. There are two hypotheses, the New World and the Old World hypothesis. Of course, there is a lot of debate between that, and there has been a lot of colonization and recolonization events between the Old and New World. The common ancestor of Cystocerca must have come some 6 million years ago, and they have been diversifying over the last 8 million years. This is the latest reference that I'm quoting here. From 7.9 million years, they have been, for the last 7.9 million years, they have been diversifying. And Cystocerca belongs to Cystocanthacrididae, the subfamily Cystocanthacrididae, which has got 35 genera. And in the Old World, it is represented by only one important genus, that is the Cystocerca, whereas the majority of them are present in the New World, the Albinus and Cystocerca genera, which are important, which are more in the Old World, or here we have just few genera. I'll come to that. See, the Cystocanthacris group, we have the Nomadacris and the Valanga species, as well as the tree locust Anacridium, all belong to the Cystocanthacridinae. They are few species which are present in the Old World or in our country, in the Indian subcontinent, whereas a majority of them are present in the New World, in the South American place, which you can see from this Bayesian tree based on molecular data given by Hojun Song et al. In 2017. They have put up this one. See, the majority of the Cystocerca species are in the American continent. Then coming to the evolution of density-dependent polyphenism, it is reported by Hojun Song that earlier the ancestral Cystocerca were having the behavioral plasticity, and they did have different color marks. Then they changed to sedentary grasshoppers. There was a loss of behavioral plasticity. And once again, there is a regain of behavioral plasticity in some, whereas some have the loss of the color plasticity, and they show as sedentary grasshoppers, and the others are the swarming locusts. So this particular typical character has to be shown by a locust to be defined as a locust. So this is the evolutionary history given by Hojun Song, a very nicely worked, perfectly well done work over them last few years. Then the gregarious phase is one which is most important, that matters a lot and causes a lot of damage. And outbreaks have been reported. The last outbreak, which came in May 19 or May 19, and were up to June 20, it was there. So in Kenya, it has been reported for after 70 years. In Somalia and Ethiopia, it has been after 23 years. And in India, after almost 26 years, since May 19, it was reported after 26 years, this locust upsurge had come. So the reason, all of you knowing that it is because of the increase in the serotonin content, and of course it has been artificially tried also in the laboratory conditions. It was tried and it was proved that just by rubbing of the hind legs or touching of the hind legs, and the smell and sight of the other locusts promotes this activity, and the serotonin content increases, and they become from solitary, so they change over to the gregarious behavior. So these things are very well known. Then coming to the desert locust recession area as given by the FAO, because the FAO with the Locust Warning Organization are the prime organizations who are working on this, who are keeping a constant watch, and you can see the recession area. This is a very well known area, and the limits are the dashed lines, the dotted lines are showing the invasion area limits to the maximum where they can go. Then desert locusts have been appearing periodically over a gap of 2 to 3 years or 5 to 7 years. And in India, 26 locust attacks have taken place, but after independence, we consider it as 25 attacks have taken place. And among all these, the most serious ones were in 49-55, in 62, and in 1993, with more than 167 and 172 swarms coming over. And the latest attack was in 2019-20. But one very important thing to notice was that they are only during the summers, but the swarm this time took place during the winters, and that was a very big challenge. The winter swarm posed a great challenge, according to the Deputy Director of the Locust Warning Organization in India. He said that yes, it is really a matter of great concern. So tons of pesticides were used to control them, and luckily they have been able to control. There was, although metallism had come in, but in the African, FAO has reported metallism and Isoclay has shown very good results. But so far in India, we are yet to, I think, come up with good data and good results on that. We can try, and I'm thankful to the NBAR for having sent samples to our people here in Rajasthan. They have tried it, but I'm unfortunately I do not know what happened to the results. But they should have got some encouraging results, I think. But we have not been conveyed, we have not been informed what results they have got with the NBAR samples of the metallism that was sent. It is really a good start, and NBAR has got a lot of scope to work on it. And I think a coordinated effort can be done. Because of the COVID, people could not come. Otherwise, had it not been for the COVID, I think it would have been successful. We could have made a joint effort. This was the global situation in May 2020, which was shared by the FAO people and the locust warning organization with us. And you can see the swarm, and the predictions made for up to July 20, it was made. And fortunately, we were expecting in September 20 also, but fortunately it was controlled and nothing came up. So we are happy that it has been under check. Then this was the situation in May, June 2020, and you can see it went up to Madhya Pradesh, Indore, and even I had received photographs from Kerala also, and it was locust, of course, but there it should not have caused serious damage because it is all immature stages only. They have to migrate, and they have to come back to their original breeding grounds, which is in Rajasthan in the Indo-Pak border. This is the dotted line showing the breeding ground where they have to come back. They cannot breed in other places. This is the only place where they can breed within the Indian subcontinent here. And of course in Yemen. So the border area is very, very important. And when in 99 we made surveys, we had to involve the border security force. There were a lot of organizations which are to be involved in the LWO, the FAO group, and since he was a FAO scholar, so we were permitted, and the border security force came to our help. And the student was not allowed because he was a foreigner, and I was asked to go. So I went along with them to the border areas. We surveyed that area in 1999. And so there are a lot of restrictions. So locust working has to be done only in this particular prime area. And I would suggest that any studies, any biological study, whatever studies we do, should be within this area so that we come up with very good data rather than doing it elsewhere. Other things will be only in laboratory conditions or simulated desert conditions in the laboratory, which will be very much costly. That has to be taken up. Then this is a locust swarm, which is given by DNA India. There are a number of photographs were sent to me, locust swarm, for telling that yes, is this a locust, or yes, we had to tell it is desert locust only. Then the three locusts, see the migratory locust, this is a solitary form which has got the raised pronotum, whereas in the migratory form of the migratory locust, this will be flat, it will not be raised. The same way you can see here the desert locust with three different colors. This is the immature adults, and the down below is the mature one, which is almost egg-laying or has egg-laid. And here on the left down, you can see the Bombay locust, the Patanga or the Nomedocris group, which we have two species here, Nomedocris japonica and Nomedocris succincta. Japonica is few, but succincta is important, it comes over in large numbers. These are the three locusts which are in Indian conditions, which we can see in the Indian subcontinent. Then again, here I am trying to show the laboratory reared ones, which we had reared here recently. This was the desert locust and the migratory locust. You can see they remain in the solitary phase. These are the solitary ones. Then we prepared a chart showing the difference, because there was a lot of difference, confusion in the identification of desert locust and migratory locust and the Patanga, that is the Bombay locust. So we made a chart showing the major differences that, see, these two, the desert locust and the Patanga and the Bombay locust are Psittacantheridae, and they have got a prosternal tubercle. Whereas the migratory locust is a Idiopodini. It does not have it belongs to Idiopodini subfamily, it does not have the tubercle. The very significant difference is you can see the tubercle. If the tubercle is not there, it should be a migratory locust. If at all it's a locust. So, and in the case of the desert locust and the Patanga, you can see the differences in the tubercle. Then you can see the differences in the genitalia, male genitalia particularly. You can see the lateral view of the male genitalia and the ventral view of the male genitalia. It shows a clear-cut difference. You see W-shaped structure or the cut portion here in the desert locust, whereas it is not there in the case of other locusts. And even in the female genitalia, you can see the major differences. So this kind of a chart we had prepared and we had submitted also. I think I had sent a copy to the NDA also. Then these are details showing of the same thing, which I'm showing in detail. You can see the clear-cut differences in the desert locust. Then in the migratory locust, even if you see the mesosternal lobes, here it is angulated. In all Citracanthocrididae, it will be angulated, whereas in the Oedopodini, it will be rounded. It's not angulated, and it is wide apart. There are a lot of differences. We need not go in for molecular analysis and all that. Just simple morphological taxonomy does a great work. Then coming to the Nomatocris saxenta, that is the Bombay locust, you can see again it is a Citracanthocrididae, so the mesosternal lobes are very acute, but the poststernal tubercle is different, and the subgenital plate in the males, the ventral and the lateral aspects, you can see it's a lot different from the desert locust, and the tibial spines will be black tipped, whereas in Citracanthocrididae satavica, which is confused with this, the hind leg tibial spines, tips are crimson red, not black in color. They are crimson red in color. So there's a lot of difference. These are the photographs that I received for identification from April to June 2020. People sending Citracanthocrididae satavica, then Aularchis afraid that it's becoming it's a locust. When they see people see in large numbers, they confuse. Then somewhere from Punjab in the fodder crops, Aedelis abruptus. Of course, Aedelis abruptus and Aedelis senegalensis, it is a common pest problem in Senegal. In the African countries, it is a problem. It is, though not a locust, but it is almost like a locust. It's a problem. But in India, we do not have serious problems of this Aedelis group. They are Oedopodini group. They are the Oedopodini subfamily. Then from Tirunelveli, Valanga nigricornix was sent. Someone confused it for a desert locust, but it is not a desert locust. It can see the hind wings details. It is a clear-cut Valanga nigricornis. There are other species of Valanga also. Then the pyrgomorphid, the calotropis one, is also again somebody got afraid that in large numbers, anything in large numbers should be locust and sending that. So this is a pyrgomorphid, Piculoceras pictus. Then in our study, what we had done, we had tried to study in 1999 about the desert locust. We used the solitary locust grasshopper, this locust hopper from 3rd instar to 5th instar. We give different food. This is Cenchrus biflorus, Herba persica, Brassica oleracea, then Ricinus communis, castor, then guar, then Arachis hadis, groundnut, then weeds like tribulus, then pearl millet, and then mixed food. You see, mixed food, in mixed food, the development period was the lowest, taking only 17.5 days in the mixed food. So it took very little time to mature into the 5th instar. And the development period according, see the 3rd instar, 4th instar, and 5th instar, it is the 5th instar which takes the maximum time, whichever food it is. Whatever kind of food we give, the maximum time taken is in the last instar. And that is a well-known fact. Even the FAO reports are showing the same thing. And in mixed food, the development period took very little time from 3rd, 4th, and 5th instar, because I have not taken the 1st and 2nd instar because they're very difficult to catch and they are very, risky. You can kill them so we have to be very careful. So we studied from the 3rd, 4th, and 5th instar. Then we also studied that, these host plants affect whether they transform from solitary to gregarious phase. In bui, that's a common weed there, it remained in the transient phase to solitary phase. Whereas in a burret, that is the thorny topper type of a plant, it changed from transient to gregarious. That is Cenchrus biflorus. This is Cenchrus biflorus. In that, it changes from transient to gregarious phase. Likewise, in cabbage, castor, and cluster bean, it remains solitary. Whereas in groundnut, feeding on groundnut, it changed to gregarious phase. And in kanti, it changed to gregarious phase. Whereas in pearl millet, remained in solitary. And in mixed food, again, there was a chance to change from the solitary to transient and to gregarious phase. It was a very short period of study. We could have continued it. We should have done it to give more emphasis on that, to know that mixed food also contributes from the soil to change over from the solitary to the gregarious phase. Whereas there are certain food plants like groundnut, it does affect. And that is why cluster bean in most of the desert area plant, arid zones, they grow cluster bean because it is less preferred. They say that it's less preferred because it does not convert into solitary from solitary to the gregarious. That could be one of the reasons. Well, then this is the, I thank you all for joining me, and this is the last picture of the most, now should be conserved. It's a species which is under almost extinction group. I would say it is we find very rare of this teratodus species, Teratodus monticolis, particularly the hooded grasshopper. They are few, and once again, I thank you all for joining me and the NBIR and the ICR for giving me an opportunity to share my views on the locusts and the proper identification, which causes a lot of confusion also. And some important aspects upon taxonomy. We should continue, and we are all of the opinion, particularly as retired persons, we would request only for some small funds to keep continuing. So 1 or 2 persons to work with us, and we keep on continuing, keep up with our collections. I have 1 lakh of collections of insects with me. Now I'm afraid how to leave it alone. Now, because although I'm retired, this 1 lakh connection should be maintained, should be kept up. That is my purpose. That's my goal. I wish it should continue. Thanks a lot.
Yes, sir. Thank you, sir. Thank you very much for the elaborative talk on the Orthoptera diversity and also the locus, ecology, behavior, and also the most importantly, the diagnostic characters. Sir. There are a lot of confusions in the locust diversity, and also they're misconceiving the other species as locust, and then there's a panic among the public. Yeah. So this will clear a little bit of doubts, at least in this color of audience. We are thankful to you for bringing out the diagnostic characters and also explaining the brief aspects of different groups of Orthoptera. Thank you so much for your nice presentation, sir. We'll just look in if any questions are there. Actually, there is one question from Saranya, that is the pest-resistant plants, whether there are any pest-resistant plants that are efficient to control pests.
Yeah, well, see, somebody has to work on it, particularly in the case of hieroglyphs group. We had studied it. There is a local, millet, which is called as kangani. When kangani is grown along the borders with the maize, the grasshoppers prefer to feed on kangani rather than maize. They don't like your hybrid maize and all these improved varieties of maize, they go for kangani. So that way a farmer can safeguard. And then cultivating marigold to a great extent.
Sir, your audio is not there, sir. We are not able to hear you, sir. I think we are not able to hear you, sir. There is from Telangana, they would like to contact you for the identification of acridins from Hyderabad. It is from Dr. T. Uma Maheshwari, who is my senior from Angru. And she is presently working as Associate Dean for Sirsila College at PJT SAU. And she would like to have a collaboration for the identification of the acridins. And Uma Maheshwari ma'am, we are happy that you are attending this webinar. Thank you, ma'am. Sir, I think we have lost
Dr. Sridevi, we can take the questions later.
Yeah.
I think now we can start with the young scientists.
Yes, yes. Okay, sir. Now we'll just go ahead with the presentations or view and opinions of the young researchers from NBIR and also from IRI and from JEDSI and also SAUs. We have 7 young researchers who would like to express their opinions and also share their views. Here in this juncture, I would like to thank Ramamurthy sir. Ramamurthy sir is scheduled to be here for this webinar, but then sir has got some important assignment which is unavoidable. So sir has sent his best wishes to this webinar and has given his PPT slides to Shashank, and Shashank will be putting forth a few points which Ramamurthy sir would like to express for this webinar, and Ramamurthy sir was instrumental in holding this webinar also. So we are thankful to him, and we'll call upon Shashank to give his presentation and also his views.
Thank you very much, ma'am. I think I'm audible.
Yes, 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 Shashank.
Yeah. So I thank the organizers, and I'm really fortunate because Ramamurthy sir was busy. So he suggested me to put forward his points. Actually, I'm going to present very quickly because we are short of time on how the NPAB has taken shape. Because I'm sharing my screen. I hope it is visible to everyone.
Yes, it is visible, Shashank. Go ahead.
Yeah, thank you, ma'am. So, yeah, I'm really happy to cover this topic. Actually, when yesterday Ramamurthy sir and Dr. Sridevi ma'am asked me, I was not prepared. I thought I will put my experience with NPAB, and I will explain only about the work which I have covered and what are my problems. But when sir told I will not be available, you just have a brief about NPAB. So I thought what I have to explain, because NPAB is such a huge project, and I personally thought, except the taxonomists, many of the people they are not even aware what is this project about. So today I'm going to talk, most of my slides will cover what actually NPAB has achieved in his 2 terms or 2 plans, that is around 10 to 11 years. So I put forward this title, reminiscing ICR network project on insect biosystematics and a way forward. Coming to the conceptualization of the NPAB, I always, when I joined IRA in 2013, I was new for this project. When I was discussing with Ramamurthy sir, he used to tell me, so this was just happened because so many good people are come together. And it all started with a lead center at IRA, and the person who is Ramamurthy sir himself, who conceptualized, and he put forward his views with IC Council. And during that time, the council was really supportive to understand the importance of taxonomy, and they brought all these institutes, including the present lead center, which was NBIR. And all these centers have been selected. For example, Aligarh Muslim University, BHU, Tripura University, which are traditional universities. But people have seen the expertise, taxonomic expertise in these fields. If you take Aligarh Muslim University, they are wonderful legendary work in Hymenoptera. In case of BHU, Dr. Raghuraman, who worked on Aterigotes. Like similarly, every institute have been selected based on the expertise. For example, University of Agricultural Sciences, because Dr. Viraktamat sir was there, and like that. So it is well conceptualized project, which includes at the beginning around 17 centers. Later in the second plan, the non-performing centers have been reduced. So around 12 to 13 centers have been kept till the end of this project. And the important objectives are the first one is exploration and collection of agricultural important insect biodiversity, which includes mites also. This is very important because many times we neglect other important biotic stress, which is also mites. And the second objective is biosystematic studies, which includes identification, morphological characterization, and documentation of insect pests along with natural enemies. Third objective, which they have included, is development of bioinformatics tools, inventories, and fourth objective, development of multimedia-based diagnostic aids. And the fifth objective, and which is very important, and I personally believe many of us who are present in this symposium got some help from this project, that is especially the objective, which is human resource development and capacity building in biosystematics. And the glorious start of this NPIB, it happened in 2005, 2006, but the active period is from 2007 onwards till 2017, which was being discontinued. In the first objective, what they have done is, like, I'm mainly concentrating on the surveys which have been done from IRA and some other institutes also. So you can just see the coverage. So they have covered like more than 4,000 localities in the entire period of 10 or 11 years, which covering almost all the areas. And that's a huge collection or survey data which NPIB has generated. And the methods which have been used in NPIB, all the methods from different centers, they have used all the methods which are standard procedures which we use for collection of insect pests. And also, especially from IRA, they have evaluated different light traps for analyzing which are these the light traps which are more efficient, especially which wavelength is more efficient. And even though a simple study like that, which had so much of data from 2017 to 2011, there are about 113 observations of data, observations they have taken to produce so much of efficacy data of these light traps. And when it comes to collections, most of the collections which are done from 2007 to 2012 comprises the following orders, around 15 to 16 orders. There was an intensive collections, both physical collections by using field surveys, by using sweep nets and all, around like 4.5 to 5 lakh specimens they have collected during this period. And only by light trap, there are about 9.5 lakh specimens which have been collected in NPIB. And you look at the crops which are covered. See, many people, what they think is, oh, it's a taxonomy project, just whatever insects they will collect, they will just describe. This project is highly focused with respect to the mandate of ICR. That means the coverage, there are about more than 150 crops have been covered in surveys, and everything is meticulously planned. The crops which they have to cover and the stages which they have to go for surveys, and all those things have been meticulously planned. And these are the few examples of crops which they have covered in the project. And every field survey, the associated scientists and researchers, they have documented digital photographs in the field. You might have seen the websites in NPC, National Postal Collection website, are the famous websites of NBR, the fields data you might have seen. So many people, I know that in agricultural universities, they will use for teaching those images. Okay, it has been done over a year of 10 to 11 years continuous surveys and continuous work through NPIB. And only at IRA, we have taken about more than 10,000 field images. Okay, that's the amount of data we have generated, only the observations which are taken in the field. any taxonomy project is evaluated mainly based on the species which we describe. Okay, so every time many people, a taxonomist will always be evaluated how many number of species he has described, how many number of revisions he has conducted, how many number of genera he has done. So this project alone, which helped in describing more than 480 species. I think Prashant Mohanraj sir at the beginning only told about around 500 species. This is far ahead with any other organization who are continuously working on that particular group. And also the main advantage of this project is a lot of diagnostic keys have been developed and a lot of catalogs of particular group of insects have been developed in this particular project. These are the few examples which I'm showing you how this project has helped in advancing the taxonomic research in particular group. For example, this is a new species, new genera and species of whitefly which has been described by Dr. Dubey, who is a whitefly specialist. Till that time, the whitefly taxonomy is only mainly based on simple microscopic studies. But because of the addition of scanning electron microscope in this project at IRI, Dr. Dubey started understanding the structures of the whitefly using scanning electron microscopy. That led to development of so many new novel characters for describing the whiteflies. Similarly, in many groups, for example, while describing Viraktamatiya Srinivasa, which is a new genera and also a new species of Coleoptera, there are so many new characters which have been studied and which helped in describing all these groups. And there is one very famous species in ICR, that's Mangalus bicapus, that is dedicated to our previous Director General, Mangal Raiji. So this species is present in IRA campus only, but nobody has studied till an exclusive acarologist came in this project as a SRF. So that means it was really so much of species which are not really explored have come, have shown up. We are present in this world. Okay, and this project has having very aware of. Project also helped in developing so many illustrated keys for higher taxonomic levels up to species level. These are the few examples of taxonomic keys which have been developed from NPC, IRA. Just a screenshot I have put about few keys for Lepidoptera and Hemiptera. These are very useful tools for educational purpose, especially for masters and PhD students and undergraduate students. Like this, many illustrated keys have been developed for up to genus and species level. This is a key which is developed by Dr. Shakti, who is now a scientist at Delhi Naturalist Museum on whiteflies for identification of different genus of whiteflies. And the facilities and the expertise from which are developed from this project, which helped in redescription of known species very comprehensively. This is a paper which is published in one of the European journal. This is a very common species. You might have seen Phycotis radiata on any ficus plant, especially the garden ficus. But this paper has included so many characters. You can see this is a biology, the first plate, and next the larval ketotaxi they have included, genitalial adult morphology, later pupal morphology, and so many characters. Okay, and also they have reported parasites which are associated with that. So this project trained people and to do a common work very comprehensively. And also many novel species have been described, but those species have been described with lot of data. For example, as I previously also I told, but there is another example which is a Diphoronia dacariensis, which is a psyllid, which is very common psyllid, wherein which this species, it has been redescribed, but with lot of scanning electron microscopy photographs. During this time, and also this project paved the way for molecular taxonomy in India. Even though molecular taxonomy was there, but there was limited funding in ICR for molecular taxonomy. And there is so many, this is one of the study which is done by Dr. Ashokan sir and group. There are so many molecular markers, species-specific molecular markers, which have been developed from this group. And I really thank NPIB for initiating such kind of work way back almost 8 to 9 years back. One of the important classical example which have been solved through NPIB, because I'm covering 1 or 2 examples which really help directly to understand how taxonomy helped to solve the issues in IPM. So one important issue is Bemisia as a species complex, wherein which many of us know there are so many biotypes. Now people have designated at this point as different species. Like 5-6 years back, when these studies have been conducted, and this was the first study under NPA which has been conducted and analyzed that they have done a lot of surveys. You can see throughout India they have collected whiteflies, Bemisia tabaci, and they proved that Asia 2-1 and Asia 1 are very dominant except throughout India. However, there are few biotypes like Asia 2-7, which is only restricted to Delhi, and Gujarat, this Miam 1 and other biotypes which are only restricted to Gujarat. So these are really helpful study during that time. And also the expertise or taxonomic expertise which has been developed through NPAP helped in contributing in other projects. For example, we had this project along with Ranchi lac insect on lac insect, wherein which we have used so many characters to characterize lac, taxonomic characters, molecular characters, and the chemistry of the lac, the composition of lac, and also the endosymbionts which are present in lac. And this led to description of so many new species in lac insect. And Dr. Aisha, who was expert in this group, she worked with both the project NIAP and also NPAP, and her the facilities which have been developed in NPAP, which also helped contributing to other projects. And also we have studied the in-depth the host-plant relationship of lac insect and also the host Simelata. This is very interesting study which are already all these papers have been published. And just to highlight the one, this established the stylet penetrability of the lac insect up to phloem tissue, and which are the tissues, the stylet, which are the tissues which are being concentrated by lac insect. And the next part, which is everybody appreciated in NPAP, is development of bioinformatic tools and inventories of insect biodiversity. The famous website I will always recommend to many people is from NBR, which is crop pest website. Many people use that. I think more than lakh viewers are there for that website. But before that, the biological control agent website has been also developed by NBR with the help of NPAP. And at IRA also we have developed so many digital tools, but most of them at that time they were in CD-ROMs. Okay, so we are now we are planning to update them as a websites. And another important part is development or capacity building. Only almost around 50 or 55 RA SRFs have been trained under this project. Around 12 PhD theses have been awarded, and many RAs, they have attended international conferences. At this point of time, among these people, around 15 people have been placed in institutes. You take ZSI, Silk, Central Silk Board, ICFRE, SAUs, and also in ARS. So, so many people who trained under this project are placed in these institutes. And under this, there are so many trainings we have conducted, around 48 trainings we have conducted, and around 32 contractual staff also trained in taxonomy. And another wonderful service which already present in National Pusa collection through our IRI is insect identification service. However, the addition of NPIB has enhanced our what I can say improved our count with respect to the species we identify annually. You can see before the onset of NPIB, we are on an average we are identifying about 2,000 specimens. However, when NPIB came to IRI, so it suddenly jumped to more. It's a 50% jump. We have almost sometimes in 2011-12 we have identified up to 7,500 specimens because of the trained manpower which we had during that time. And just a calculation, if you take the economics, because most of the policymakers, they talk about economics of taxonomy. If you just calculate the rates charged by Naturalist Museum London, just average 2,000 specimen, if you take around 5 crores annually, we can save from only identification service. Just keep aside other taxonomic contributions which we have done in NPIB. And the major aspect in taxonomy is literature. During now, most of the literature have been digitized, so it is very easily accessible. But during 2007, 2010, it is very difficult to get literature. So NPIB has been instrumental in scanning all these literatures. There are about 13,000 pages have been scanned under this, and most of these soft copies are available. And many people, they just okay, there's so much of the things people have done, how much budget it has been allotted to NPIB. I was also wondering what is the budget. So when I looked into this, it is for 5 years, it is around 16 crores. This is, and I personally felt this is not a huge budget. Okay, easily any government agency can offer if you are producing such a wonderful contribution. And the major crux of NPIB is infrastructure development. Infrastructure development with respect to IRA, like in NPC, you can see what are the changes it has done to NPC, especially the introduction of mobile racks and all, and also lot of microscopes, molecular lab we have developed, scanning electron microscopy facility we got, auto montage, so many equipments it has been procured through NPIB and which has been used by many institutes. And how I have benefited, this is about NPIB, how it has contributed. When I have entered, when I joined IRI in 2013, the first project I'm associated with other than in-house is NPIB. So I'm really fortunate enough to work with Dr. Ramamurthy sir, because he guided me to just begin my research in taxonomy. During that time, lot of common agriculture insects or lepidopteran used to come. So he just told me, so we have this insect identification service, you just start IDing them up to family level, because as a beginner, nobody will believe what you ID. So he just told me to ID up to family levels. That's how my journey into Lepidoptera taxonomy started. So the first thing I started is insect diagnostics. Later, I got myself into the Lepidoptera taxonomy, and I've been involved from last 7 to 8 years in many taxonomic revisions for new species discovery and resolving few phylogenetic debates, monitoring and management of invasive pests. I'm fortunate enough that because of identification system, I got into reporting many invasive species because it's an advantage at stay. If you're doing some service, yeah, people will knock your door and they will ask you, please help us in identifying and reporting these species. And at the same time, I started learning about how to manage insect collection and how to digitize them. And I'm really fortunate enough because the major area of taxonomist is research expeditions. If you don't go out to field and you don't collect your specimen, you can't really publish anything. You can't sit in your lab and you can expect somebody will give you specimens. You have to go out and you have to work. To go out and work, you should have a continuous funding facility. At the beginning, I'm really fortunate enough to get funding from NPIB. Later, there are so many funding agencies I could tackle. So these are the two main area I want to just present in before.
Shashank,
Yes, ma'am. I'm just finishing.
Yes.
Yeah. So the last two slides. So these are the few minor contributions from my side from last 8 years. And what is post NPAB? Why NPAB was so important? These are the major concerns which, as a youngster, I experienced, because when NPAB was there, okay, you've got some constant funding for your field collections and your research and for some molecular work. But when you lost some constant funding, you have to manage your collections and also you have to do your research. So I'm fortunate enough to get funding from DST SARB, especially under organism and evolutionary biology. And that MOF stopped funding through ICOPTEX. There are still open, but there are very limited extramural funding by ICR, but most of our research is funded by in-house projects of ICR. And I'm ending with a statement. I thought when with project NPIB fostered in which was before, and conducted and ignited basic taxonomic research which influenced the pest management strategies, and instrumental in building national and regional infrastructure for insect repositories. You may take any repository, maybe NBR, maybe IRI, or maybe TNU. It helped in some way or other. And support from ICR was immense, and hope it will reinitiate such kinds of projects to keep taxonomy alive and kicking. Thank you everyone for listening, and I thank organizers for giving time.
Thank you, Shashank. We are actually running.
Yes, ma'am.
Thank you for giving the elaborate view of the NPIB functioning and also the benefits that we have accrued due to the NPIB network project. Now I request Ankita Gupta from NBIR to give her views and opinions. You have to unmute, Ankita.
Ma'am, I'm just sharing the screen.
Yes.
Is it visible, ma'am?
No, yet to share. Yes.
Visible, ma'am?
Yes.
Yeah, I'll just I'll be, in a nutshell, I'll be talking about our institute, and I thank the organizers, and I greet all the respected seniors, peers, and the viewers here. Since the time is very short, I have just put the concrete what we have and what we have achieved as NPIB and what we have lost as NPIB project has gone, and what we are as a bureau, and where do we stand nationally as well as internationally. So to talk about this, we have a galaxy of very dedicated taxonomists here, which I have just put on the screen on the left and the right side. I would just like to, most of you must be knowing, but it is my duty to introduce them. The head of the division, Dr. Joshi, Dr. Sridevi ma'am, Dr. Shalini, Dr. David, Dr. Mahindran, myself, Dr. Patil, Dr. Sampath, Dr. Rachna, and Dr. Om Prakash. Now, as you can see, we are covering many taxa. Dr. Joshi is covering aphid, scales, and mealybugs. Dr. Sridevi ma'am is covering scarabids, Dr. Shalini pentatomids, Dr. David fruit flies, and many flies in general, Dr. Mahindran weevils, myself parasitic wasps, Dr. Patil EPN, and Dr. Sampath spiders, Dr. Rachana thrips, and Dr. Om Prakash Navik, trichogrammatids. So these I have included the taxonomists who somehow during their course of studies, whether they were PhD students or they were young researchers or they have taken up their career, they have been directly or indirectly, they have been benefited by the NPIB project, including me. So whether it would have been our joint expeditions, or we would have shared the specimens, or we have got the reference collection which has been made during the NPIB project time, or whether we have shared the expertise, or we have got the influence and the motivation from our peers, and most importantly HRD, because we ourselves as a team are part of the HRD which has been generated, most of us, by the NPIB project. So during the NPIB project time, we did get the infrastructure facility which we had built upon, and during that course of time we have been accredited by the Ministry of Environment and Forest and Climate Change as the National Insect Museum for agriculturally important insects and mites. So we have been upgraded. And just to highlight what is our potential as a group of taxonomists, which is contributing a lot towards nation biodiversity exploration, you just can see the team of 10 of us have contributed in the last 3 years with new species, a whooping number of 120 new species and genera. We have offered identification services of around 2,000 services to different organizations. 5 research monographs we have given. Checklist is 5 and keys are 25. We have developed e-databases online 4, which we have added, and new records more than 200. Now, this is just when we have very, very limited infrastructure with us. Sorry, infrastructure in the sense of funds and manpower. Now, if I just portray this reference collection, what we have at the Bureau, what in line with Dr. Shashank, what he has told positive about the NPIB project, you can just see during the time of NPIB project, how we have flourished as a museum. We, till 2017 March, you can see we had massive increase of the specimens. At least it used to go about 50,000 specimens also. But just after the removal of NPIB project, we had a steep drop in our collections, which is really a loss to the museum as a whole, where we do stand. Though we have almost 2 lakh specimens now, which are dry mounted and preserved. But then if you see, our additions are not more than 10,000 specimens in a year, and which is a very sorry state at present if we consider us as a national museum. Now, why do I emphasize on that? Because of the limitations. The limitations in the sense is that we lack systematic sampling because of our fundings. We cannot go for systematic samplings. We do not have permanent collection permits like ZSI has, and many other organizations, they have permanent collection permits. And most of us, because of fund shortage, we are only concentrating on alpha level taxonomy, though we have much more potential to do it, but then we are dependent on other divisions like the molecular division for the supporting data. We ourselves do not have a unit where we can do the molecular stuff with us. And what we face is that majority of the cases, even if we are doing molecular studies as a whole, as a team of taxonomists I'm talking here, if you see the reference collection in the NCBI database of the Indian specimens also, if you go and if you want to do your elaborate studies and you search for the vouchers, many of the places you do not find the vouchers on which we can refer to. So with the limited resources and manpower, most of us are working as one-man army. We go for collections, we do our work, we do our sorting, we do our identifications, and in the end we end up publishing with lots of rejections many a times, which is not very encouraging as young taxonomists. And like other countries, we do not have a team of parataxonomists or the volunteers which help us in the museum, especially in the case of the bureau. We have a curator which is about to retire this year. So this is the fate of her collection. So the scope, what I mean to say is that even though we have limitations, we have been performing to the best of our ability. We have potential. We aim for advanced integrative studies, long-term biogeographic and evolutionary studies. And we do help in multidisciplinary approach. And we also need to, like we have been benefited by the project, we need to develop second line of taxonomists so that in future there is no gap between. So with this, I would just like to end my words, and the utmost take home message which I would like to convey is that without a proper continuous funding, there can be no scope for taxonomy in this country. Thank you, madam.
Yeah, thank you, Ankita. Thank for that crisp presentation. Now, without much delay, I'll request Naresh Mishram from IRI to present his views.
Yeah, good evening, everyone, and thank you, organizer, for giving me this opportunity. I will not take much time. I will just share my experience regarding my leafhopper taxonomy. I have described around 63 new species. I have one monograph, and I have done some revisions in this group. And also I have added some around 100, more than 163 DNA barcode generated for the leafhoppers. Now, during entire my journey, like when I started this my leafhopper taxonomy, my mentor Dr. C.A. Viraktamat, he helped me throughout till he is helping me out when I am doing my taxonomic things. Now, even though during the journey I felt some of the problems which I wanted to share with you. Some problems are generalized problems. Some are like particularly technical problems for those who are the youngster. They should realize when they are describing the species. So first of all, the generalized problem I can tell you, we are about to come up with one monograph that is already published in the Silidini. One tribe is there, Silidini. It was in the fag end of publication, and there was one query from the reviewer. So they wanted that one of the genus we synonymize, they're asking us to look at the type material, and then you take the decision. Now, the problem was that the type material was in California Academy of Sciences. And Viraktama sir told me, no, we need to confirm it from the California Academy of Sciences. At the fag end of publication, they have taken 3 months to give us the correct answer, or to give us the reply from them that, okay, what you have done is right. What I mean to say is, if the accessibility of type material is fast, like it should be digitized, so we can get those images and we can confirm those, so we could save this time. Another important thing is what I wanted to mention here is the maintenance of type material. So we need to inculcate in our youngsters that the importance of holotype, how to treat the holotype. I have seen many of the students, when they are handling or describing the new species, they are not taking utmost care. So what I used to do, I used to send my student to CAV sir lab, and the way he used to handle the holotype, so they should learn how precious it is. Because when I was examining the holotype from certain institutions, those holotypes are damaged, not properly taken care of. So at least we need to inculcate these things in our youngster. Another important thing is, I always discourage the student to go for isolated description. And also I will urge the youngsters not to go for the isolated description of the species when there was no revisions or monographs are available. If they are going with the isolated description of single species, when there is no revision, it will lead to the synonymy. Until and unless you will have the revisions, don't go for those isolated description. Another important point I wanted to say here is the funding for the taxonomy. We are getting the funding from DST, MOEF, as well as DBT. But the problem is, when they are giving the funding to us, they are giving very less amount for the collection trip. But as a taxonomist, we need more money in the collection trip. So we need to, just I wanted to put forth here so that we can take up this issue with the other organization also. And the last problem what I face is the difficulties in extracting the DNA from the museum specimens. So we all are here, some molecular biologists are here, so we should come with some kind of protocol so that we will get the DNA from the old specimen what we have here. Thank you, madam.
Thank you, Naresh. Thanks for your thought-provoking points. Thank you. We will now request Devanshu Gupta from ZSI, Zoological Survey of India, who is also working on Coleoptera scarabid beetles. I would request him to present his views and opinion.
Thank you, ma'am. Thank you for giving me the opportunity to present in this in this conference or webinar. Dr. Sridevi ma'am, thank you very much. Dr. Distinguished entomologist, Dr. Chandis Balal ma'am, Dr. Sunil Joshi sir, Dr. Ankita ma'am, my friend Shashank, and madam. Madam has told that to tell some idea about what we are doing in Coleoptera in ZSI. I will be dealing with this beetle diversity in India. What we have, since 3 years, and my guide director of the ZSI, Dr. Kailash Chandra sir, has carried out so far. Challenges and gap areas and way forward in Coleoptera taxonomy, being in charge of the section. With few points, I would like to tell that ZSI has cataloged so far 1 lakh species we are having in our country. Out of them, 5,600 have been described exclusively by ZSI scientists. And we in our collection are having more than 50,000 species, which have been, most of them have been digitized, and all the type material also has been digitized, as we have 12,000 type specimens. I will be briefing these things, but directly coming to the detail. So far, insects in our country are concerned, so far we have 66,911 species of insects in four different classes as per 2013 classification. This Collembola, Protura, Dipleura, and Insecta. So Collembola includes 345 species, then 20 species of proturans. Most of them are from Kerala only, and dipleurans. And insects, we all know that they are the most diverse group with 26 orders, and the eight insects order that are Coleoptera, Lepidoptera, Hymenoptera, Diptera, Hemiptera, Orthoptera, Thysanoptera, and Odonata. They constitute the 94% of the total insect diversity of the country. The other orders are very few in number. This is the total diversity we can say. Here you can see the Coleoptera itself is 22,334 species, is leading in all the groups. So these species are classified in 114 families, and here in India we are working on very few families. Madam and I are working on scarabaeids, and we have recently collaborated in the Sericini subfamily. I am working here, and madam, we are publishing together also. And Ramurthy sir is working Corcolinidae, and very few families have been treated in India so far as Coleoptera taxonomy is concerned. I will not be giving detail of all these numbers. So far, Indian diversity, out of the 4 suborders we have in the world, 3 are reported from India, 176 families, 114 have been reported from India, and 22,334 species. This number is what we have cataloged so far, with the synonymies and combinations and their distribution in states and biogeographic zones, all the taxonomic changes we have cataloged, and we are going to bring this document soon on Indian beetle diversity. These are some pioneer works in Coleoptera from Indian region. Now, coming to the beetle diversity from different time, like Carl Linnaeus has described 50 species from India. Then in 1998, Sengupta and Paul estimated that 15,500 species have been recorded. Then after that, in 2007, estimates were made by Ramakrishna and Alfred Saab in 17,431. Now, after this, all these things, these were all estimates, no checklist or catalog of entire Coleoptera was with us. Now, what we have reached to the conclusion that there are more than 22,334 valid species till date are present in our country are described so far. And these species we are, I am saying, or we are saying that based on our catalog only, what we have prepared in ZSI. I would like to compare how much diverse is in case of beetles is our country, that you see the Australia, then this Palearctic region, 80,072 species are known, and you see our India's diversity. And North America also so much rich, it is having 25,000 species, but you can see our diversity. In the Palearctic region, you will be surprised that out of these 22,000 species, the Himalayan Palearctic region is including more than 10,000 species are known from Himalayas only. Then coming to the endemic beetles, there are more than 3,100 species of beetles which are exclusively found in our country. Out of the total endemic invertebrates, which are 26,782 species. This is a summary of what diversity we are having in our country. Archoestimata is still not reported from our country. Myxophaga is having 7 families from India, 7 species from India in Hydroscaphidae and Sphericidae. Then Ediphaga, which include this carabates and Dytiscidae and that is the Dytiscidae. This Scarabidae is the leading in Ediphaga, more than 2,645 species. Then Polyphaga, like all we know that Curculinates, Scarabates, all these groups are highly diverse. These are the 7 major families which are most diverse in India. Not only in India, and Stephanoidea, I will say, is one of the largest family in entire animal kingdom, with more than 56,000 species so far described. Then Chrysomelidae, Carabidae, Curculinidae, Scarabidae, Ceramicidae, and Letridae, and Tenebrionidae. These are the major, these families majorly represent the Coleoptera of India. Then coming to the beetle diversity in different ecosystems of India, this data we gathered from our catalog, what we have prepared from agroecosystem, 359 species which are pests and predators in of different crops and used in as predator. Then freshwater ecosystem, we have 776 species pertaining to 17 families, 17 aquatic and semi-aquatic families. Hydrophiloidea, then entire 6 families of Hydrophiloidea and Dytiscidae, Noteridae, all those families. In soil, this is very surprising, 8,862 species are directly or indirectly associated with the soil, and out of 22,000, over 20,000 species are directly in the forest ecosystem, we can say. Not man-made ecosystem. Only 340 species have been directly associated with the exclusively with the mangrove plantation, not associate plant, but true mangrove plantation. And half of the diversity is present in Himalayan region, 10,533 species. Cold desert, very few species have been reported, that is 93 species, and from desert area only 326 species have been reported. But this is very good to know that island is having in India only 0.25% of land, but you see the diversity which is here, overall diversity, it is 8.5. Means islands like Andaman and Nicobar Island is quite rich in diversity of not only of beetles but all other total fauna also, like 11,000 species are known from Andaman and Nicobar Island. This is what we did in biogeographic zones of India. Previously, we were thinking that Deccan Peninsular region will be having the much number of species than the Himalayas, but we come to the conclusion that Himalayan, that is the northwest, west, and central and eastern Himalaya, excluding northeast. We have not, in this classification, we followed this Rogers classification of Wildlife Institute of India. 10,533 species are known exclusively from Himalayan region. And there are no such record from coastal area of beetles from the coast. And from peninsular India, 3,325 species have been recorded. These are I am not detailing all those things, just because we are having very few time. This is the same thing, you can say. And also we have assessed the diversity in different states, political boundaries. can see that this Sikkim area is having the highest diversity, West Bengal and Sikkim. Most of the descriptions have been made from the Sikkim and Darjeeling area. Like 5,000 species, more than 5,000 species have been recorded from this area only. And very few work have been done in Chhattisgarh, MP, and this Rajasthan, then Gujarat, then this Andhra Pradesh and area, and then Odisha, then Jharkhand. Bihar also very few records are present. But this West Bengal, Northwest Himalaya, Western Himalaya, and Northeastern area, Nagaland, Mizoram, Manipur, Meghalaya, they are also very less explored in case of beetle diversity. These are, I have made it, but I will skip these slides because of time. These are two families of Heteroscaphidae and Sphaericidae which are reported from India. Then Edifaga, this Carabidae is the largest family, the ground beetles and the tiger beetles. Then these Gyrinidae, Heliplidae, Noteridae, and Titicidae, they are all aquatic families. We have good expertise of these two families in our section. One person is Ravi Kushwaha is working in Carabidae, and then Shri Sri Kumar Ghosh is working on the aquatic families. Then Hydrophiloidea also we are working, aquatic group. We have six Hydrophiloides families from India. Hydrophilidae is the largest family. Then Hysteridae, nobody is in India is working at this stage, but we have 260 species from India, and in Jadesai we have very good collection. In these Staphylinidae, I told previously one scientist was working in Jadesai, Mrs. Abasar, but now she has retired. This group has been completely left out in our collection. We are not yet focusing, but I am planning to engage somebody, some student to work on Staphylinidae, because this is very diverse group to work on. Then Scarabeoidea, I am dealing with Scarabeoidea, and good number of 2-3 students are doing PhD also on this. I am basically focusing on Geotupidae and Scarabids, Rutellini, and this Melolonthini. Like madam, we have recently collaborated one project on description of with Dr. Doug Ahrens on Sericini. Scutioidea, nobody in India is working on these families. We have 98 species so far reported. Most of them are described by scientists abroad scientists. Then Dactylidae, nobody is working. We have 17 species from India, and we have the collection in ZSI's our Coleoptera section also. Then Buprestidae, 604 species of this family are known. But pure taxonomy of this group, nobody is, means no descriptions are coming from Indian scientists. Then Viroidea, this also is a completely lacking group. As far as taxonomists are concerned, 207. Nobody is but we are taking this group this time. One student I have taken CSIR JRF, I have given this group for Viroidea. 217 species of this group is known from India. Elatidoidea is quite diverse family. 816 species are known from India, and this group also is not that much worked out. Then Dendrodontoidea, 8 species are known. Then Bostichoidea, this group has been worked on in FRI, and Dr. Vijay Veer sir has worked a lot on this group. 191 species are known from our country. Then Limaxilloidea, 6 species are known. Most of them are from these islands. We recently collected from Nicobar Island. Then Claroidea, 334.
Dr. Gupta, I think because of time.
Yes, sir. I will sum up.
So what we are looking for is what are the difficulties you are facing, and what are the things which we can put up to council.
Sure, sir. Yes. I am summing up, sir. I will skip all those things. Just I will come to the what we have more than 3,14,425 specimens in our collection of beetles only. Out of them, only 54,000 are identified. So I invite, and out of and nearly 4,835 species are there in our collection with 76 families. I invite the scientist in this forum, expert on any family of beetle, you are welcome in the section, and work on any group. We will facilitate and help us in identification of different families. What we are working, we can do, but on the families which nobody is working, or if anybody is interested to work on particular family, and I, on behalf of the Director Zoological Survey of India, invite you to collaborate with us, work with us, and we will, if you come to the our section, or we will provide you the facilities, everything here in ZSI. Then the main lacunae what we are having from, we have so far only sequence 50 barcodes, but this thing we have to enhance in our research in Zoological Survey of India in Coleoptera, especially in Coleoptera, but in other groups like Thysanoptera, Lepidoptera, and spiders, DSI has done quite good work. These are some recent publications what we have made. Then the main focus is why I have shown all these beetle diversity in India and all those things, because very few, in what we say in Hymenoptera taxonomy, you say most of the work have been done by Indian scientists only, like parasitic Hymenoptera, Dr. Hayat sir, Dr. Narendran sir, Dr. Ankita ma'am, and many good people. But in case of Coleoptera, what we see, when I catalog the specimen, most of the species have been described by, not by us, not by our people, and the specimens and the type specimens are in British Museum, and we are not in that much condition to identify those. And in Coleoptera, what I feel, very few people are working, especially on the taxonomy of India. I like to point out some people like Thomas Sabu sir and Ghate sir in Maharashtra, Madam, me, Ramurthy sir and lab, Dr. Mahendran sir. Very few people are working in Coleoptera especially. So lots of species are yet to be described. We need to enhance the capacity for young researchers, especially PhD students and researchers, to work in. And I welcome any student or anybody, if he or she is interested to work on beetles. We will try to, if for as for interest, we will try to give try to accommodate as JRF or SRF as PhD student in our organization. So far we can do. Like we need this catalog or complete checklist of Coleoptera, especially for India, that we have done it, and we will be doing it soon. And the taxonomy, especially fauna of India, need to be published on various groups like genera, different families. Then we need to assess the species diversity of beetles species, especially the indicator species at micro and macro level, and new species and new records for these are, and monitoring of groups, monitoring of long-term monitoring of these. We have identified the species, described the species, that's okay, but how it is behaving in the environment and what is its impact of climate change on that species, that is also need to be assessed. And this our DNA barcoding also. Then then first thing I will say that synergy between all the institutions like IRI, ZSI, and like we have started, madam, and we recently started working on sericine beetles. Like we can collaboratively work on many things, and the collections what we are having and the collections what other institutions are having, we can share the information and in collaborative mode and work. And also the habitat suitability modeling of especially the pest and predator species, that will help us in this to understand the how the species beetle is behaving in the environment. Okay, this is very small team what we are having in our Coleoptera section under the leadership of Director ZSI, Dr. Kailashandra sir. Then division in charge, Dr. Raghunathan. I am working on scarabaeids. Sri Ravi Kumar Kushwaha is working on carabaeids, and Sri Sujit Kumar Ghosh is working on aquatic families. And recently, we have 4 PhD students who are working on different families, like PhD, one in scarabaeids, then one in coccinellid, then Devika is working on sericine, Priyanka on coccinellidae, Jojit on scarabaeid, and Soumit is, we have recently joined as on chrysomelid beetles. Thank you. Thank you.
Thank you, Devanshu. Thank you so much for giving the overview of the activities that is being carried out in ZSI and
Thank you, ma'am.
also
Thank you,
giving an
ma'am.
overview of the beetle diversity. We also place on record heartful thanks to Kailash Chandra sir,
Thank you,
because
ma'am.
we have also extended our invitation to Kailash Chandra
Yes,
sir
yes,
because
ma'am.
of such a busy schedule and also other commitments. Sir couldn't make it to this webinar,
Yes, ma'am.
but sir has sent, Kailash Chandra sir has sent his best wishes to this webinar, for which we are highly thankful to Kailash Chandra sir, and also thanks to you, Devansh.
Thank you, ma'am. Thank you, ma'am. Thank you. Thank you, ma'am.
Now we'll move on to the next young researcher, David from NBIR. David,
Yes, good evening to everyone. Thank you, the organizers, for making me part of this to express my opinions and view regarding the insect systematics, importance, challenges, and way forward. I'll just quickly finish because the time is running out. So I am basically a taxonomist working in NBS since 2011 till date. And my area of interest is taxonomy of Indian Tephrididae, where I've described several species of fruit flies and several new distribution records I've dealt with. So I could come to taxonomy because of the major pillars in taxonomy like Dr. C.A. Varakthamba, Dr. V.V. Ramamurthy, Dr. Purni, K.D. Pratap, and all these taxonomists had molded me what I'm presently. Because of that reason only I'm able to speak before you. And in Tephrididae, I could contribute anything because of whatever the help I've taken from the experts, foreign experts like Dr. D.L. Hancock, one of the authorities on Tephrididae in the world, and Dr. Friedberg, Dr. Kornev, Dr. Norberg, and Han. So just I would consider that I am still a taxonomist. I have not molded it into a systematist. So we are still into the state of alpha taxonomy, and we have too far to go to reach the systematic stage. When Dirk had presented, Dr. Dirk had presented, we came to know that how far behind we are regarding this status of the systematics aspect in India. So the significance, of course, we all know that because of the enormous diversity, the traditional taxonomists, those who have been working in the Indian fauna, had classified the species into different groups of organisms, different classified into different taxa, like whatever they to solve the enormous diversity they have classified. So our purpose as a systematist or a taxonomist, our purpose is to make the classification quite natural. That is, it should be, it should not be artificial classification. We have to elucidate the synapomorphies or the shared derived characters with which we can define a group. So biological species concept is the most popular species concept, though. But presently, the thing is that we cannot, in the dead specimens, most of when women were able to study the how they are, whether the process of the reproductive isolation we may not be able to study. In such situation, phylogenetic species concept arises, and under that gained more significance due to the publication of several papers which has come in very recently regarding the phylogeny of several groups. So phylogeny gives more importance. That way we have to step
Dr. David, you are not audible.
Hello.
Now
Hello, sir. Am I audible now?
Now you are audible.
Okay. Sorry. I think, sir, here internet is a problem. Sir, internet connection is not there. I'm using my data card. So that's a problem. So the major challenges with regard to insect systematics is that main...
Switch off your video. Switch off your video. That will be fast.
1 second, sir. Yeah.
Yes, go ahead.
So my perception is that because those who are became expert in the field of taxonomy, they doesn't have any problem for having a good expertise or contact with the taxonomic experts. For the youngsters, those who are planning to come into the field of taxonomy or systematics, the primary difficulty one may face is that contact with the taxonomic experts. Unless they come to know that we are really genuine in this group, it will be difficult to get an rapport with them and come so that we can exchange our ideas, so that we can exchange the literature and get more idea about a particular group. So that's what we learn. And the most important difficulty which I face personally is the examination of the type specimens. Those which are abroad as well as within India. In India also, we have faced several problems like that to get a specimen. And moreover, I personally feel that those who are taking care of in the museum, they should encourage, because unless a specimen is studied, unless a specimen is dissected, it is very difficult to understand or study what that species is, how to study the phylogenetic relationship. And in the case of examining specimens type from the museum from abroad, so there will be several times there will be a problem where we have to convince the customs fellow. Sometimes it will get halted there. We have to convince them that this is a specimen which has come to identify for us to identify. It is not a something to deal with the National Biodiversity Authority. So all those issues may pop up when we have to work upon systematics aspect. And access to original description is another difficult aspect which we have taken in, which is very difficult in case of insect systematics. And regarding % of species CO1 will always give a weak phylogenetic signal compared to other markers. So in order to have a thorough or a valid or robust phylogeny, we need to undertake several other molecular markers like 16S, 12S, and all those things has to be incorporated to get a full picture of the, to get a strong phylogeny tree to elucidate the synapomorphy, or to come out with a clade which is monophyletic. And as Naresh has already mentioned regarding the difficulty in getting the DNA extracted from the museum specimen, that's one of the major challenges which we face. And of course, the Dr. Peter has mentioned about the immature taxonomy, the difficulties. So in fruit flies also, there are lots of gap in immature taxonomy that has resulted in not resolving the phylogeny of fruit fly. Fruit fly is one of the economically important groups. The phylogeny, the higher level phylogeny of that group has not been fully resolved yet. And definitely coming to we being aspiring entomologists or taxonomists, we feel like we publishing our journals, publications in high rated insect systematic journals. So there, once we submit the paper only, then we'll come to know how far behind we are regarding the aspects of systematics. So they expect us, their level of standard is comparatively different from what we are working upon. So that way we have to work more particularly regarding the integration of the molecular aspects, biochemical, biological, and morphological. All these things have to be integrated to get a full resolved tree, so that we can come out with a valid decisions or valid points regarding this taxonomy systematics of a particular group. And particularly when regarding the collection aspect, we can make out the difficulty in accessing the protected area. So if I have to go for a collection in a particular state, I have to give a separate application, and for some other person means you have to give a separate application. So in that such situation, it's it is very, if it is come to a situation that being a national institute or ICR institute, if scientists, those who are working into taxonomy, if they are getting a permission to collect the insects from protected areas, so that will help us to document the biodiversity of India to a large extent, so that we can resolve the major problems in the identification, as well as we can strengthen our phylogeny, data regarding the systematics. And so the way forward, I would like to feel like the funding to visit various museums. So if we get one, because most of our holotypes have been deposited in several European museums, so it will be difficult for us to go and personally examine because through image also we can see, because most of the specimens we need to dissect the specimen and see it and examine under the microscope. Then only we can come to know that whether whatever we are describing it is a new or something different. So, and strengthening the molecular diagnostic markers. So we have to depend upon several markers, that capacity building has to be done, and extensive field collection in India, thanks to DST and all. So that DST, because we are getting project, because of that reason, we are able to move around and we could collect and we could improve the collection in India. And to the youngsters who are participating into this forum, I just want to tell that passion is the most important aspect in case of taxonomy or systematics. Unless you have passion, you cannot do anything in systematics, because this is I feel like the technology driven era, because all will ask for what you have worked upon, where the technology, what you have done for that. So in such situation, passion is the most important toward taxonomy. And in insect systematics, whatever you have to achieve means so many comments may be there regarding what we have achieved. Such situations, the encouragement towards the taxonomy is most important. Those who are really talented, those who are really into the taxonomy should be encouraged, so that this science be alive always and could be able to contribute towards taxonomy and insect systematics. Thank you.
Thank you, David. Thank you for putting forth your challenges and also way forward in a very crisp manner. We have two more. You can exit from your screen, David. We
Okay,
have two
madam.
more youngsters. Kalesha Swami, I think you don't mind if I admit Saili, because she is draining out her Wi-Fi. It seems she has sent the message. So I will request Saili to present her views and opinions. Then we'll go for you, Kalesha Swami. Saili, are you there? Saili, yes, Saili. You can present. To share the screen, there is a green button at the bottom of the screen. Shared screen is there. You go there and then share your screen.
I think her name is not in the panelist. Just check.
She's there, I think, sir.
So if it is there, you tell her there is a green button,
just
share
check,
screen.
Check. Yeah. She is put in the chat, sir, how to share the screen.
No, if it is not, she is not panelist.
She's not there among panelists.
If she is not panelist, she will not get that option itself.
Okay. Then
I have added, ma'am. I have added.
You have added. Okay. Thank you, Shashank.
Ashik.
Saili, you can briefly tell about. Okay.
Yes, ma'am. Am I audible to everyone?
Yes.
Yes. So good afternoon, everyone. And I, first of all, would like to thank Dr. Sridevi for giving me this opportunity to express the challenges I faced during my work and still facing. And I really feel good that this platform is available so I can share my experiences and get inputs from you all. So I will be talking about taxonomy of Indian water beetles in brief. So in brief, as Dr. Gupta has already told about Coleopterans, it's a very huge group of insects, and several features are there that this group is evolutionarily very old, and they have colonized the water for several independent aquatic invasions and diversified into 4 different subfamilies. Suborders. And to date, there are 30 beetle families with 13,000 aquatic representatives, which are adapted to live in or on the surface of water. So broadly, these are different types of freshwater habitats. Lentic, which are stagnant water bodies. There is also diversity of water bodies we have to take into account, because beetles have adapted to such aquatic ecosystems. So there are broadly, inland water can be classified into freshwater and saline systems. And these are different types of habitats where beetles can be found, like very shallow pools to very deep aquatic ecosystems. So I am representing here different types of horizontal water bodies. This is horizontal. And again, we have phytotelma, where beetles can be found in such different habitats. So why I'm highlighting these different types of habitats? So these type of habitats are less explored. So these need to be explored. So this is vertical ecosystem of water beetles. So this is a seepage, water seepage. This also needs to be explored. And lastly, here we have a saline ecosystem. So we have to study both freshwater and saline ecosystems. So that data needs to be generated. So in comparison with world, where are we? So in India, there is a platform developed by Dr. Wazirani. He has a huge contribution for water beetles. And with the available facilities, he has developed a huge data on beetles. However, there are some lacunae I identified during my PhD work. So the literature which is developed, which is already available on water beetles, is now need to be revised because it is for 50 to 50 years back. I would also like to highlight Dr. Sujit Kumar Ghosh, who also has contributed tremendously in water beetles. So what is lacking here is descriptions of species, like the updated literature with documentation of digital documentation. Then now newly updated distribution data is lacking. And also I found similar problems of colleagues here, that museums, that is well curated museums are needed, well curated specimens are lacking. Then secondly, access to the museums where specimens have been deposited. So I also face the same difficulty where I don't have specimens which are well curated to compare my specimens with the already described specimens, because most of the holotypes are deposited in international museums during the British period of India. But also I also have experienced good way of collections where I visited GKVK through my guide Dr. Hemant Ghate. So his colleague Dr. Virakta Math is a well-known scientist in this institute. So this one of the museums I visited, so I could access these specimens. So as to nullify the lacunae of updation of literature and also exploration of different areas, we studied Northern Western Ghats. These Northern Western Ghats are known Western Ghats Sri Lanka biodiversity hotspot. And these are the physical features. I won't be going into details as we are short of time. So these are the physical features of this area. As we all know that altitude is the major contributor to have the gradients of environmental variables or climatic conditions in the Northern Western Ghats. So as altitude varies, there is a gradient of precipitation and temperature, which are important for distribution of the aquatic beetles. So as a method, what we did as my PhD work, we collected specimens and majority of them are in ethanol. And then we studied in detail internal and external morphology, using different, these I have mentioned here, different imaging system. And we also perform scanning electron micrographs to fill the lacunae in the literature. Then again, another lacuna was to the specimens are deposited in different international museums. So I contacted and collaborated with museum curators and experts through my guide, and visited this museum where I could understand how specimens are maintained. And then I learned preparation of illustrations. So this is how we try to document the beetles and their important characteristics at Modern College under the guidance of Dr. Hemant Ghate. Here is our publication where we tried to fill the lacuna of availability of important diagnostic characters through scanning electron micrographs. This beetle can be found in such ephemeral pools, literally roadside pools we can find this beetle. But this beetle was not known from Maharashtra, so we updated the record. And male-female differentiation can also be highlighted, can be seen here. Here we also described two new species of genus Copilatus, which is present in all different continents except Antarctica. But importance of this group is they are morphologically very similar, like it is a homogeneous group. And these two beetles couldn't fit into the available literature, both from Indian workers and international experts. So we contacted Dr. Yuri Hajek from Prague Museum, and under his guidance and my supervisor's guidance, we published 2 new species from the Northern Western Ghats. We also have updated the distribution of known species and update different species from the new records to the state, as well as the 2 new species. So distribution data, what I told about, that there was a lacuna of distribution data. So we are trying to cover those lacunae or rectify those lacunae.
Sorry to
Here
interrupt
is
you,
another
Saili.
new species.
Saili, you can move your slides fast, because we have some more things to discuss.
Yes, this is the second last slide of my presentation.
Okay.
Yeah.
Yeah.
So this is another habitat I explained, the vertical seepages. These habitats also need to be explored. So here we used integrated approach, where we also use morphology as well as molecular work. So this is how we perform the molecular morphology here. Interesting thing is, all water beetles have a very dense swimming hairs on their legs, and in this beetle, which is found on seepage, this beetle was lacking all the swimming hairs on its leg. So this is an adaptation to that. This can be an adaptation to that vertical ecosystem, which is semi-aquatic. It is not totally aquatic. So with the 16S and 16S rRNA and COI gene, we also have shown the placement of this new species within the microditis of Indian microditis. So this work was published in the Oriental Insects. And lastly, in collaboration with Dr. Martin Fikacek from the Prague Museum, we published another review of Silostoma. This group is really very huge. Has colonized different habitats during its evolution. And in the similar way, we tried to cover and rectify the lacunae from the Indian subcontinent of digital documentation and descriptions of new species, as well as the distribution data. So here is a map. So I would like to acknowledge my supervisors and mentors, my colleagues, especially my colleagues, friends and family, without which the fieldwork was not possible, and different institutes for grants, facilities, and Maharashtra Biodiversity Board for permits, and finally and importantly ICAR for giving me this opportunity for giving me this platform for presenting my work, so I can share my problems and get some inputs from you how you tackle the similar problems. So thank you for patient listening and the opportunity you have provided, ma'am.
Thank you, Saili. Thank you for your putting forth your work, and also what are the challenges that you have faced during the work. And thank you so much. And in this context, I would like to thank 2 stalwarts and seniors in taxonomy, Viraktamat sir and also Ghate sir. They both are inspiration to many youngsters. And Viraktamat sir has sent message when I have sent this webinar flyer to seek his blessings and wishes. Congratulations for, I'm just reading sir's message. Congratulations for organizing the webinar on insect taxonomy involving several stalwarts in the field from India and abroad. I wish you all the success. I'm sure it will enthuse talented youngsters to get into the field of insect and mite taxonomy and develop the field further to understand these organisms better, so that we can harvest benefits of their services and get more insight into their conservation for posterity. With best wishes, Chandra sir, Viraktamat sir has sent. And in a similar way, Ghate sir also has sent me a mail wishing a grand success for this webinar, and sir has extended his support. Actually, sir has wrote one very good paper in 2013 itself on the Indian taxonomy, its challenges, and what can be the way forward. So if interested people approach me, I can forward that paper. It's a very nice paper from Gate sir, and Gate sir is helping me in cerambicids, and we have published it together many papers, and I'm very thankful to Viraktamat sir, Gate sir, and Ramamurthy sir. Ramamurthy sir also has sent his message, though he's not physically here. Sir has told that his wishes and presence is there with us for this webinar. So acknowledging them, I would like to now invite Kaleshwar Swami to present his opinions and views. I mean, for this session, he's the last youngster.
Thank you. Okay, am I audible, madam?
Yes, audible.
Yeah.
Audible.
So thank you very much.
Yeah.
No, in fact, when Shashank was presenting, I felt very happy that I want to share one important thing is that I am also product of NPIB, in fact, and mentored under Dr. C. Viraktamat, and I thank Dr. V.V. Ramurthy as well. So, in fact, I started with termite taxonomy during the NPIB project, which I continued after coming to Shumaga, where I started with one project funded by DST Young Scientist project, and later actually I got one more project under funded by MOEF. So I'll be talking about what is this present status, challenges and way forward in brief. And in fact, we know in India around 338 species distributed throughout India, and in my lab we have collected around 60 species by now. And now we have a robust identification volumes we are depending on, published by our predecessors like Ronwal and his students and worked on at ZSI. And in fact, with those keys, we can identify. We know that termites are social insects. So if we have imagos, we can identify only up to families. Okay. Otherwise, for very few, we have the keys for genus and species, and workers we have only for family, and that means so we have keys for only soldiers. So that is the one important taxonomic impediment we have. So unless we have the soldiers, so it's very difficult to identify to species level. So I list some of the important difficulties which we face in case of termite taxonomy. So identification is entirely based on soldiers. Okay, so that means when you go for a collection, if you don't get the soldier in a colony, so you cannot identify to species level. So that is the one important problem. And another thing is that the identification is entirely based on measurement. Okay, and if you have a single specimen, for example, so you will measure, say, for example, a character called mandible index. If it fits to 0.68, so or 0.69, and it's very difficult in another character, in say, for example, head length, it actually moving to Odontoceras obesus, and that time it's very difficult to delineate and say that it is actually this species. So unless we have more number of specimens, at least 5, okay, it's very difficult to say that this is actually a particular species. And many times, say for example, if you get the soldier, you can identify two species. That means if you are really targeting to identify and develop keys for the adults or for say workers, once they emerge and leave the colony, okay, it's very difficult to associate. That means, so if you want to develop keys further in the future, you need to go and see the emergence, especially which occurs during swarming season in the evening hours. We need to collect there itself, and we should associate. So that is actually the one of the area which actually I'm targeting. And another problem we encounter is, so many of the termite species live as inquilins in other termite species. Okay, so especially microtermus, which you usually live with other Odontoceras species nests. So if you get only the workers, it's very difficult to say that which species you collected in that particular colony. So this is another problem which we generally encounter. So then, what are the solutions for these taxonomic impediments? How will you target? So of course, we are now targeting or moving towards molecular taxonomy. And in case of termites, the molecular taxonomy has been exploited already in higher classification, and lower classification is always open. Okay, so in case of higher classification, so we know that if you refer the recent modified the classification which we are following, we don't have a order called Orthoptera. Okay, so the reason being, which was merged with order Blattodea. So a wonderful publication in the year 2007, and after 5-6 years, we could agree that so the termites are in fact social cockroaches. So death of an order published by Inward, and we now everybody in fact agree that there is no family order called Isoptera. So they are actually merged with Blattodea. This actually led to some of the concerns among taxonomists. We were actually not ready to accept this change. We were actually used to Isoptera. So what were those concerns was actually with respect to the terminology or the orders which were used so far cockroaches. So what is that we used interchangeably, synonymously for Dictyoptera, Blattodea and Blattaria. So now it is resolved with molecular taxonomy. Dictyoptera means which includes termites, cockroaches and mantids. And Blattodea includes cockroaches and termites, and Blattaria exclusively for cockroaches. So if the second question arised was, what is the status for Isoptera? So where it has to be placed? So then that question was addressed in fact. And if you see the phylogenetic analysis, the termites are actually grouped, clustered along with a family of cockroaches called Cryptocercidae, which has got a sole genus called Cryptocercus, which are actually the social cockroaches. Okay, so they have got a considerable parental interaction. That actually important reason for placing the termites along with the cockroaches. And another thing is that you see here, termites are very close to Cryptocercus are very close to the termites. These cockroaches are very close to termites than the other families of cockroaches itself. Okay, so that means they diverge from Cryptocercus and become separate lineages. Okay, so now we call the termites as infraorder Isoptera, or rise to one more level called epifamily Termitidae. So both can be used. So what is the application in the lower taxonomy then? That was actually in the case of higher classification. In case of lower classification, so how good is to delimit the species based on measurement? Say, for example, one has got this much head length, we call it as one species. If it has got another head length, we call it as another species. So in fact, the caste actually development occurs based on the hormonal balance in case of the social insects. What may be, if happens to be, say, environmental influence, if these morphometry varies because of environmental influence, what we should be, then shall we call it as a different species or same species? So in fact, we try to address this problem in two cases, which I will take as an example. Okay, so one case which I actually try to explain is identification of invasive termites. So we talk a lot about invasive insects now. So termites are also becoming invasive. So in fact, we regularly get the termites from quarantine stations. So one of the vials we got was Orthoptera testaceus. And this is the measurement of our specimen, 1.25 to 1.3 in case of head length. This was another publication. You can see the variation. And this was the original publication in Linnaeus, where the head length was actually 1.38 to 1.5, which is nowhere linked to the type specimen. So that means if you decide only on the morphometry, we may consider it as a different species. So under that condition, what should we do? So in fact, if you refer the old literature as well, so many of the couplets actually are very confusing, and measurements in fact overlap, and it's not actually taking to what exactly where it should take. So of course, we had a problem with the species which was actually collected in quarantine station, which is called Orthoptera sojeti, and we could do molecular analysis, and we could able to identify that they are actually the two different species which are not being reported from India. Okay, so probably they are maybe may occur in the mainland as well, not just quarantine stations. So which we published recently as first interception of Orthoptera from India. So another case which actually highlighting here is Pseudocapritermus. We have only one Pseudocapritermus species, Plenary, in South India. So recently, in fact, we got specimens. You see here what happened. So this is the original Pseudocapritermus Plenary, which is published with a head length of 1.4 to 1.8. I'm taking only one character here. But if you see our specimen 1.3 to 1.38. So this is the Plenary and another the specimen which we collected. You see the head length and head length of this one. Okay. So which was actually very different. We can easily, the morphological evidence, we can easily publish it as a new species. But what we went ahead with the molecular analysis, and where we found that only 3 nucleotide variation in these two vials which we collected. And so we did not go ahead with the publication as a new species. So the morphological variations do occur in very conserved character, and that should not be confused as a new species. So that's what happens. So there must be many synonyms which are going to happen if you adopt the molecular taxonomy in the case of termites, which we are actually targeting. Okay, so future directions are developing reference library of different genes for all the Indian termite species. Okay, that if you do that one, definitely there must be a reduction in species diversity in India, so to an extent of 100. Okay, and another is actually say if a species is there in Karnataka and a same species which is there in Northeast India, maybe I've got a different head length or various morphometric character that may be published previously as a different species. So probably it may have happened in case of species distributed in different parts of the world. So hence there is a need of collaboration with international organization and check the morphometric character as well as the molecular character, and then phylogenetic analysis has got a lot of potential. Okay, so if you see that this was the first slide I presented. So out of 338 species which are distributed in discovered in India, so only for less than 35 species, NC5 accession numbers are available. That means, so we are not good in molecular analysis of all these termite species. So there is a greater need of molecular analysis of and also different genes. We need not to just restrict to COX gene or MTCO1 gene. So there is a need of 3 or 4 genes. So in order to generate phylogenetic relationship and resolve the termite taxonomy. So thank you very much. So I thank Dr. Sridevi and other senior taxonomists from NBAR and different organizations for giving me the opportunity to present my work. Thank you very much.
Thank you, Kalesha Swami. Thank you. It was a very brief and then very comprehensive work and opinion on the termites and also the termite taxonomy, where it stands, and also you have given a brief overview of the morphological and molecular distinction also. Thank you, Kalesha Swami. You have been support all the time. Now, sir, our director, sir, now we'll be taking up the plenary session with our experts. Chandish ma'am is there, and also Ramani sir is there, Swaminathan sir, Dr. Peter, then Sunil Joshi sir. Sir,
yes. We are already exactly 1 hour beyond schedule.
Yes, sir. Because the presentations are very good, so we are about to hear to them. Sir, we will first hear to Chandish ma'am's view and opinion, and also followed by Ramani sir. Then we'll take up any other action points are there from other panelists, sir. Okay.
Nice to see you, Chandish.
Same here, Bhaktavatslam. Excellent webinar. Let me congratulate you and Dr. Sridevi and the whole organizing team and all the taxonomists of NBR for the HOD of taxonomy section, and all of you for the very nicely conducted webinar. The invited speakers were brilliant, and I think not only the young researchers and students, all of us have benefited through the presentations by Dr. Peter, Dr. Dirk, and Dr. Swaminathan, Prashant. Everything was brilliant. And not only that, the rapid presentations by the young entomologists, that was really very, very nice, excellent. And they have really brought forth their constraints, which I think should form a part of your proceedings. Very clearly they have brought out the issues which they are facing. But then I think the highlight of today's webinar is the positive attitude indicated by DDG Crop Science about reviving the NPIB project. I was extremely happy about that. I only wish the rapid presentations were made in the presence of DDG and ADG, which brought out everything, all the constraints were brought out. Probably it would have been good. Maybe you can think of some other opportunity.
No, madam, he is very, very busy.
I understand. Maybe other occasion you
also,
can.
He said 9. Actually, we wanted to have it on 28th. 28th, he said, I am not free. Then we made it 29th. Then he said like that. He is very busy.
But then it's clear that he is for it.
That YP should have been there, but he also has sent me that he also has, because he is also in a ADG FFC. As I had already mentioned, Dr. S.C. Dubey has joined now from today. I called him, but he is little, I mean, not stabilized. Yeah, just
I
joined.
see, I see. Okay, okay,
Ma'am,
okay.
we'll be taking up the document, ma'am. We'll be
Okay,
picking up
okay.
all
So,
the youngsters.
I think also the old NPIB document, the highlights and the issues faced because of the sudden premature closing. Maybe that is what is required. And one point which I wanted to tell taxonomists, as important it is to exchange dead specimens, I feel it is equally important for live taxonomists to be exchanged, between international exchange of live taxonomists, because I think that is what will really strengthen the taxonomic work. So probably that should form a part of the possibly revived NPIB project that you're going to. It's not only the national collaboration, international collaboration, at least one per organization. I think it would really take you a long way. And I should thank you for the invitation. I really enjoyed myself. Thank you. Thank you all. Thank you, Bhaktavatslam. Thank you, Sri Devi. Thank you so much. Yeah.
Thank you, ma'am. Thanks for your brief and crisp opinions and views. Thanks a lot, ma'am, for your presence and also for your views, ma'am. We now request Ramani sir to give your opinion and views, sir.
Thank you, Dr. Sri Devi. I would like to remain out of the video screen because of the fact that I'm also going through a data maze. So I would, I mean, if you can all hear me, it's fine. Can you hear me?
Yes, sir. Yes, sir.
Okay. I thought that this was a very well organized webinar where you had brought in two experts from two different fields who talked about wonderful beginning, especially about integrative taxonomy. I think that's a field which really need to be picked up by a lot of youngsters and several of us, because integrative taxonomy, where you are linking up various things, changes the way you look at taxonomy itself. And I think Darren did a very good job of trying to present that and to try and tell you, because by experience, by their experience, they know how, if you put in different phylogenies by using different methods, how the taxonomy of the group will change. Similarly, Dr. Peter Spek also had a wonderful presentation to talk about immature taxonomy. The congruence of immature taxonomy with adult taxonomy brings out a lot of information about how that immature taxonomy could be a good supplement, not only a supplement, but could be an independent thing to look at in terms of trying to understand how the two things are congruent. I'm sure Dr. Sipek must be having a lot of other information about how when you look at immature taxonomy and adult taxonomy, there could be a lot of differences that has been noticed in terms of the taxonomic understanding of that particular group itself. So in some instances, there could be a lot of congruence between the two. In some instances, there could be a lot of differences between the two. So when we start, this is also part of integrative taxonomy. Another aspect of this is that when you merge immature taxonomy with adult taxonomy, you could also be making it into an integrative method of trying to understand how immature taxonomy imprints on the adult taxonomy. So I think the way forward is to try and look at it from an integrative aspect and try and see how best you can use integrative taxonomy to further the taxonomic understanding of different groups. So I really think that these two presentations were very nice to try and come out with this kind of an information and encourage several youngsters who have been watching this to try and get into their fields of comfort. What is important is that to try and get out of the field of your comfort and try and understand some of these aspects so that you will be able to develop the understanding of the taxonomic this thing of that particular group. And I also would like to appreciate all the youngsters who were presented, especially Sayali, Shashank, Naresh Meshram and Kaleshwara Swami and David and others, who brought out their points very, very nicely to try and tell what are the difficulties that they're facing. And I majorly think the major disadvantage which has been is because of the closing down of the NPIB. NPIB closing down has been a major disadvantage for several people. And I think funding is the major issue. And Ankita Gupta brought out how the collection has come down because of the lack of funding. So this, I think, is very, very important. Collection is very, very important. And unless you go out to the field and look at material in the field, it is very, very difficult to do your taxonomy without understanding what is happening in the field. And so unless you have funding to do these things, it becomes very, very difficult. And NPIB gave a very good opportunity for a lot of youngsters and a lot of others to go out into the field and collect material to different parts of the country. Now you cannot go to different parts of the country and collect because of the lack of funding. So I really think that NBAR should really take this up and try and make sure that NPIB at least is functional in several institutes and several in universities and organizations which are leading in this field, so that the collection and the database of collections from different parts of the country really improves. I think it's really running. I mean, you must be really running out of time. I would like to end with that. I think it is a wonderful opportunity where you can put forth these points very nicely, because the DDG has been very, very positive on this aspect. So I think you have to really put it up to them to make sure that funding comes for people to work on taxonomy, especially to do collections and do also other aspects of taxonomy. Thank you. Thank you very much.
Thank you, sir. Thank you so much, Ramani sir, for participating and also giving the good points and also views, sir. Actually, at this juncture, I would like to place on record thanks to Ramamurthy sir. Ramamurthy sir also told that, as you suggested, sir, we'll be preparing the document and also we'll be presenting to ICR. So sir told that you send the document, I'll modify, I'll also put my points, I'll also prepare, and then we can send it to ICR. That's the promise Ramurthy sir has given, and also Ramurthy sir has extended his support to present the proceedings of this important webinar in Entomological Society of India website, and also ready to publish in Indian Journal of Entomology as a special coverage. Sir was speaking yesterday and was putting forth these points. So I'm highly thankful to Ramurthy sir, and also for you all giving these suggestions, Chandish ma'am and Ramani sir and all the experts. Now I request Dr. Peter, if you are available for a few minutes, we can take your opinion.
Okay, hi again. Hello. So it must be already dark in India. It's
Yes.
7 p.m. if I counted the time correctly. So I don't want to waste your time. I think this was a great opportunity to see the Indian colleagues, to see how Indian entomology is developing. There is a lot to do in general in entomology in terms of describing diversity and in terms of saving at least the knowledge of diversity, which is vanishing before our eyes. And thus I would love to express the only thing that we should, as taxonomists and as biologists in general, we shall push more forward into explaining people that entomology and collection of any biological samples is nothing which is threatening biodiversity, but it helps to understand biodiversity, because in many countries, and I'm speaking from the situation which is happening in Europe and situation which is happening also in other countries, mostly people who are collecting out in the field are those who are victim to the laws of biodiversity protection, which is contradictory, because most biodiversity is not destroyed by collecting for scientific samples. It goes away with agriculture, with human activities, and we all as entomologists have to make it, have to ask the governments and the authorities to enable us to study the diversity before the diversity vanishes, because we are losing such a big amount of species every day, and we shall really stand up against these bans, against these issues with national laws which are preventing us from international cooperation. But to end up optimistically, I've seen that there is a huge generation of young Indian workers growing up, and I'm really happy about that, and I'm happy to cooperate in the future with some of you, and well, Good luck to all young scientists, and I would encourage them to keep with this beautiful science. Thank you, and thank you, of course, for the invitation and for all the people here.
Thank you, Peter.
We enjoyed, Dr.
That's a
Peter,
valuable suggestion.
your lecture as well as the discussions. We enjoyed, Dr. Peter. Thank you. Thank you very much. Anybody else?
Thanks, Peter, for taking participation. Swaminathan, sir, we will have it, sir. Swaminathan, sir, any wrap-up suggestions? Swaminathan, sir? I think sir has audible issues.
Yes, unmuted.
Uh-huh.
I think he can't.
Is it, sir? Sir, then shall we wrap it up, sir? A few
Yes,
points
yes.
from your sir.
I think he's trying again.
Hello there.
Yes, doctor.
Yes,
yeah,
sir.
can you hear me now?
Yes.
Yes, sir. Yes, we can.
Wrap-up points. 1 or 2 wrap-up points, please.
Yeah, thank you very much for this nice opportunity. Really, apart from the experts like Dr. Peter Spitt and the other one from Bonn, the youngsters have presented equally very, very nicely, and I think it's really a good thing, and it gives us a boost that we have some people at least, because I was very much afraid when I retired that most of the things are not being taken care of. And of course, the NPIB closing down was also a big shot for us. It was really a loss. I hope with the DDG's positive statements, we are able to revive it, at least with a small funding or at least having 1 or 2 SRFs with us. No need of any high going equipment, but at least the running costs, if we have, we can cover a lot in Orthoptera. I still have, I think, at least more than some 50,000-60,000 species that need to be finally identified properly out of the almost nearly 1 lakh collection that we have. So I think it was a great job. Thanks a lot. Thanks to everyone, and I hope it does continue.
Bhaktavatslam, there are some questions in the chat box and question answer, whether they can deposit specimens. So probably you may have to respond in the chat box. Some youngsters are asking in the museum whether they can deposit specimens.
Yes, provided full data is given. So as you know, it is NBA, that is the 1 point also I have made. So if no other points, I just have a few points to make. Of course, I will be always fast in making my points. 1 point is that revival of NPIB. I think Dr. Joshi knows. Then see, this time EFC DDG has asked us to make a presentation. Institute-wise, he was listening, then it was going on. And under that, we have made this point that NPIBA, we also made that this is the thing which has been continuing for so many plants, it was suddenly stopped. Then he said he will revive. Then the second point also for the scanning electron microscope. And after that, then almost like 2 months, the presentations were going for all the institutions. And finally, suddenly he was asked to submit the EFC. So we made a rush up and submitted. It has gone to, as you know, it has gone to ADG PAM. There they have made constraint to the budget. They have made 15% of budget of our BE. So because of that, we projected earlier almost like 57 crores. Now we have to reduce to 47 crores. So because they are not agreeing. So whatever they have told, you have to stick to 15%. And also for each year, it should be 15% of the previous year, like that. So accordingly, we removed that, and now we have got the opportunity to again revive up. As Sridevi is mentioning, we will make again a draft, present it to Dr. Ramurthy, and again we will submit to DDGCS. Again, we will try again that, because what happened, I know you know that agrodiversity project, which was continuing in NBPGR, but they have removed NBP, our institute, NBIR and MAU. But again, DDG's intervention, we are again included, and we have projected some amount. I think we'll be getting it soon, because that has come in the PAM projection also. Again, we will take up the issue and we will see this is included under the ADG PAM. Once it is included in the ADG PAM, then it will come as a separate project, and we will try for that. I think another 15 days or within a week or so, we will make a presentation and do that. Then another thing which I wanted to make it, why don't we think of making one of the institutions, one of the museums as a international repository. I think now NBAIM is always claiming that their museum has been recognized by for international repository. I think this is very essential. Either IARI or even NBAIR, though we are in very infancy stage, we should try for that. I don't know how to make it. So we have to ask our institute, our museums also to be recognized as international repository museums. That I think it is very essential. We have to make one point about that. Of course, the other thing is sharing of voucher specimens. That is very essential. Again, as Madam was telling, international exposure is very essential. All our scientists should be trained at international museums so that they get the knowledge about taxonomy. Again, we may have to think about the collaboration with non-ICAR institutes or non-SAOs, so that it is traditional institutes. I think this, again, we may have to think about that. Again, molecular systematics, I think we have to make a strong institution, maybe either at NBAR or maybe IARI, because that is already he is very much interested. That is the future science. So when we are talking about the integrative systematics, that becomes a point. So we have to strengthen our molecular systematics, either any of these institutes. Again, I think we can have a discussion with all the senior people and make a call on that. And again, we have to find out where it can be done. And that is also one point which I thought. Then one presentation was systematics of invasives. That is very, very essential. I think we are neglecting that. That is also one point which has to go as a recommendation, that systematics of invasives which we are anticipating. Because once it enters, it is difficult. So before that, I think we should enlighten and educate our systematics people how the new species or invasive will be looking out. So these are some of the points which I have noted out. If any point is there, we will have. Then again, Dr. Sridevi will wind up with vote of thanks from you.
Yes, sir.
Can I
yes,
have?
Sir.
Yes, Dr.
Yes, you want to add?
Yeah, no, Dr. Bhaktavatsalam just recently told about the invasive. No, we are planning to have a one-day webinar on invasive pests and all that. And I wish if somebody could also, well, I'll let you know about the details, and we would like to have that very.
No, what I am telling is systematics of invasives.
Yeah, exactly. That can be also a part of that. In February itself, we are trying to organize it.
Yes, yes. Please, we will be taking part.
Thank you very much. Thank you.
Yeah, if
there is one Veena, I don't know who is that.
Sir, Veena is my JRF only, sir.
Your JRF.
Yeah.
Okay,
I have put her to take up the questions.
Okay,
and
no
also
problem.
The notes.
Okay. I thought our K Veena.
Sir, no, sir. So if there are no any other suggestions and points, I think I'll go, because I have requested Sunil Joshi sir, but Sunil Joshi sir is not able to have the audio connectivity, and sir is here with me. So on behalf of director sir, head sir, and my own colleagues from NBIR, I would like to present a brief vote of thanks. We know we are already late, but then vote of thanks is also very, very important, because all the people who has taken interest and then participated in this webinar need to be thanked for their presence, sir. I'll start with our ICR officials, with DG sir. DG sir is always supportive and encouraging such type of webinars. So we profusely thank Mahopatra sir, Director General ICAR, and we thank extremely with extreme thanks to DDG Crop Science, T.R. Sharma sir, for his presence and also sparing his valuable time and also giving insightful thoughts and giving a positive response towards the revival of network project on insect biosystematics, which is the need of our, because we are not getting any funding from any other sources other than DST, and very few from Ministry of Environment and Forest. But then if our ICR itself can fund the taxonomic activities, that will be a great boon to Indian taxonomies. So we are highly thankful to DDG Crop Science, T.R. Sharma sir. Then we are also extremely thankful to YP Singh sir, ADG, FCC, and also who has today taken charge, to whom we have briefed that such a webinar is going
Dr.
on.
Dubey, Dr. Dubey.
Dr. Dubey sir. Yeah, we are thankful to Dubey sir, who has assumed charge today. And we are thankful to our director sir, Bhaktavasalam sir. When I have gone with this proposal, sir has readily accepted, and sir has told that we'll take it up. It is really needful for the present taxonomic research work and activities to be taken forward. For giving us the right support and cooperation, we are thankful to you, sir. And I'm highly thankful to our head, Sunil Joshi sir, who readily accepts any proposal that we take to him, and he has given his inputs also, and he has helped me in organizing this webinar. Whatever small discussions I used to take up with Sunil Joshi sir, and we both together we have conceived this, and of course in consultation with Ramamurthy sir, which I'll be telling now. I'm highly thankful to Sunil Joshi sir, and I'm thankful to our colleagues in NBIR. Before getting into other logistics, I'm highly thankful to the speakers of today, especially Dr. Dirk and Dr. Peter. In spite of their busy schedule, they're having, since it is the beginning of the year, they have so much of work. But when I have communicated to them, they have readily accepted it, and for which we are highly thankful to them. And Dr. Dirk's presentation and Dr. Peter's presentation has ignited young minds to take up taxonomic research, and which is the main purpose of this webinar. So we are highly thankful to Dr. Dirk and Dr. Peter, and thank you so much, Dr. Dirk and Dr. Peter. Then we are highly thankful to Swaminathan sir. Swaminathan sir also, I have kept him a long wait, like getting dates from international speakers. So thank you for bearing with us, sir. First, I thought I'll take the dates from international speakers, Dr. Dirk and Dr. Peter, and then I communicated to Swaminathan sir. Swaminathan sir is kind enough to accommodate the time. And thank you so much, sir, for being with us and also for participating and giving a talk, sir. And Dr. Ramamurthy sir, which when I have conceived this one, first I discussed with Ramamurthy sir, and Ramamurthy sir has given the idea of including the young researchers also. First I thought we'll have the eminent speakers and then few views of quick views, but Ramamurthy sir told no, we can have their work also and what problems they are facing. So it is the idea that has come up from Ramamurthy sir, the young researchers, and that has shaped up well this webinar. So I'm highly thankful to Ramamurthy sir in absentia, and I'm also highly thankful to Kailash Chandra sir, Director ZSI, then Dr. Viraktamat sir, Dr. Gate sir, to whom I have approached also for kind tips and suggestions. I'm highly thankful to Manjunatha sir, who is always a well-wisher and then kind enough to be part of all activities of NBIR. I'm highly thankful to Manjunath sir for his presence and for his time. We are highly thankful to our former directors, Varghese sir, Krishna Kumar sir, and Chandish ma'am, for giving their valuable time and also for giving very good inputs. And I'm highly thankful to Chandish ma'am for staying till end and also giving opinion. And madam always keeps encouraging, and madam is patient enough to be there till last.
I have been seeing she sitting there only. She didn't go out.
Yes, sir. Yes, sir. Yes, sir. Yes, sir. That's what we are highly thankful to you, ma'am. And we are thankful to Varghese sir and Krishna Kumar sir also. And we are thankful to all the organizations, and we are thankful to the young researchers, Dr. Shashank. Actually, special thanks to Shashank, because we had some problems. Our NBR people must be knowing, we had yesterday the breakdown of the network. There is no internet, absolutely internet. We were very panic, and then I have pressurized Pratibha madam, and Pratibha madam was also running off restoring the connectivity, but whole of yesterday it was not there. So we were so much worried, and in between also it tripped. So what we did is we tried to have a co-host, and Shashank readily agreed, and Shashank is co-host for it. And for that there is a intermittent drop in the connectivity, but then the webinar went on well because of the co-hosting, and also he has facilitated and assisted us in live streaming on YouTube, for which we are thankful to Shashank. And we are thankful to the young researchers, starting with Ankita, David from NBIR, and Devanshu Gupta from JEDSI, and Naresh Meshram from IRA. Naresh Meshram also we are highly thankful, because he said he is not feeling well. He is having cough, but then when I said you put forth some points that can be taken up as action points and also get into recommendation, so he agreed, and then he was there with us. So thank you, Naresh. And we are highly thankful to Saili from Pune, Modern College Pune, and also Kaleshwar Swami from UAHS Shimoga. So for giving beautiful points and also beautiful overview of their work, and also what are the hiccups and hitches that they have found during the course of their work. So I'm extremely thankful to all my NBR colleagues, and especially the colleagues from Division of Germplasm Collection and Characterization, who have extended all the support in carrying out this webinar. And I'm highly thankful to Pratibha madam, the AKMU unit, because the webinar hosting and the making panelists, everything, because as a moderator, when I'm sitting here, Pratibha madam with her team, Zaida, Manju, and Sonkusare, AKMU team has given extended their support. And here I would like to mention that 2 international participants are there. I forget to mention 2 from West Africa. One is Annette, who came here to get the training, and one Koli Bailey from Ivory Coast, West Africa. So thanks to them also for participating in this one. So my thanks to AKMU, and also my thanks to all the participants who have registered, and still
almost like 190 were there.
Yes, 190 were there. The registrations were high, but must be most of them, I heard that they are viewing on live streaming also on YouTube. So those who are viewing in live stream also, I'm highly thankful. And also I'm highly thankful for all the participants, because I can see my own colleagues, Dr. Padmaja, who was our IMC member, is also present. And Dr. T. Uma Maheshwari, who is my senior and presently working as Associate Dean for Silsila College in PJT SAU is also there, and also Dr. A. Padmakumari madam from IIMR. So I can see a lot of my colleagues and also my students, and I'm very happy, and thank you for all your presence.
Dr. Prashant, Dr. Ramani.
Yes, sir. Yes, sir. Yes, sir. Dr. Sir, and I mean, special mention to Dr. Prashant sir for readily agreeing for the expert talk and delivering his excellent presentation and excellent talk. And Prashant sir is always cooperative, and I am highly thankful. And I am highly thankful to Ramani sir and Belwadi sir. Belwadi sir also, I could see sir has joined, and I think in between sir has left. Highly thankful to Ramani sir and Belwadi sir for giving their opinion and also view, and also for their presence. I'm highly thankful, and I think I have covered all, because so many things to collate. So if at all I have missed anyone, I give my excuses and sorry, but it is not intentional. I would like to express my heartfelt thanks to one and all who are involved in holding this important webinar, and my heartfelt thanks to all of you for making it success. I thank you all. Thank you.
Thank you, Sridevi, for excellently organizing, because I know for the past 2 days she didn't sleep. morning itself she came running to me, sir, I am very much tensed up what to do. Thank you, very nicely organized, Dr. Sridevi. Thank you.
Thank you, sir.
Thank you each and every person.
Thank you, sir. Thank you. Thank you, Peter. Thank you, ma'am. Thank you, Swaminathan, sir. Thank you, sir. Thank you all. Thank you, Shashank, and all
Thank you, ma'am.
researchers.
You're welcome.
Thank you. Thank you. Thank you, ma'am. We'll make a good proceedings. Thank you.